Chapter 6.1 Moringa Pests and Problems. You will find several references throughout this manual explaining how to keep your moringa trees of the highest quality possible. When we talk about quality, we're talking about as close to perfection as we can get. There are 4 fundamental elements in having a top quality moringa tree. First, we must have a moringa tree that has inherent quality. Universities and private research facilities are continuing to develop the genetic qualities of Moringa oleifera, one of 13 types of moringa. PKM1 stands for the location that particular Moringa oleifera seed comes from, which is a town known as Periyakulam, PKM, in the state of Tamil Nadu, India. They first released the seeds in 1989, improving its susceptibility of diseases and pests. Every 6 generations, they release more seeds, strengthening the gene pool. We now have the best Moringa oleifera PKM1 to select from than ever before, and they continue to be improved. There is nothing we can do in the USA to improve the genetic quality of moringa. We can improve its environment and conditions. Whether we are propagating or buying seeds, we should select the best plant material available, choosing dark and mature seeds to plant from that are loved and in good conditions, without mold, fungus, bacteria, or pest or insect problems. This chapter deals with moringa tree pests and problems. Moringa trees can have inherent problems, inherited traits that detract from the quality of a moringa tree, very similar to how a person may pass down certain traits to their child. A moringa tree with an inherent problem cannot be cured. We must strive to select moringa trees without inherent problems, or at least give moringa trees the best quality environment as possible to reduce the stress, pests, and problems that tree might encounter. The key is to grow moringa in as close of an environment of the region it originates from. They can be adapted to regions, zones, and soil types. With those changes, though, it may bring on more problems, so be aware of the moringa trees, origins, and where you are and want to grow them. Otherwise, you may be looking at more pests and problems. We must grow moringa trees under the best conditions. Chapter 3 dealt with the conditions needed for moringa growth requirements. Whenever we fail to provide the proper conditions, problems result. Problems caused by poor growing conditions are often referred to as environmental diseases. They are often difficult to distinguish from problems caused by living organisms. Third, we must protect the moringa tree from the attack of living organisms, both plant and animal, which might use the moringa tree for food, shelter, or simply damage it because it is in the way. That is what this chapter is about. Fourth, we must train and use the moringa tree in every way, without letting parts go to waste. It's too valuable. The moringa trees should look healthy because they are healthy, and every part of the tree can be healthy. That provides the beauty and aesthetic quality we want from members' farms so that we can make content and show off our practices. This facet of quality is covered in Chapter 1 with salesmanship and Chapter 8 with benefits and uses of moringa. Diagnosis or identification of the cause of moringa pests and problems is both extremely important and difficult. Fortunately, the moringa tree industry, several universities and private research institutions has already experienced mostly all the pests and problems that moringa trees would be faced with on any continent on the planet. So, we have a great baseline to work with. This chapter contains excerpts from the Florida Pest Control Guides, Insect Fact Sheets, and Landscape Integrated Pest Management published by University of South Florida, IFAS. Several books, periodicals, fact sheets, and other references are available to help in diagnosing and controlling pest problems. This manual simply provides a good starting point. Chapter 10 provides lists of excellent references and resources. This chapter is designed to assist the Your Moringa certified member, first in the identification of moringa pests, insects, diseases, nematodes, and weeds, and second in the various methods of controlling these pests. While this chapter stresses not only detailed information about specific pests, but also the cultural approaches to controlling them, pesticide recommendations are not included. Other sources should be consulted for specific applications. Pesticide laws and regulations are covered in Chapter 1, but with the knowledge from this manual, the certified member will not need to use chemical pesticides. Instead, have best management practices in place with all the parts in place for the environment to protect the moringa trees. Pests and problems are usually a result of planting moringa trees in the wrong environment or ignoring the soil first and foremost as an active approach to healthy trees with less pests and problems. Beneficial insects are not pests. The material in this chapter on insects should be studied closely. Florida has more different kinds of insects than any other state. While most are not harmful to plants, there are more than enough kinds that damage plants by their feeding, breeding, or shelter habits. The problems they cause are serious, and the damage is economically important. There are too many kinds of harmful insects to try to study and remember each kind individually. We can and should be aware of the groups of insects that commonly cause problems to moringa trees. Insect life cycle. To combat insects and pests successfully, something should be known about the way they develop and feed. Insects normally hatch from eggs deposited on or near the food supply, although in some cases, as with aphids, they hatch within the female's body and the active young emerge from the female. Adults usually have fully developed wings, although a few species of insects never develop wings. Insects pass through several stages during their development: metamorphosis. Plant bugs, leafhoppers, thrips, and grasshoppers hatch from the egg in a form known as a nymph. The nymph resembles a full-grown insect, except that it lacks wings and is smaller. It molts periodically as it increases in size, gradual metamorphosis. Moths, beetles, and flies, on the other hand, hatch from the eggs in a worm-like form, larva that is much different in appearance from the adult. The larva of a moth or butterfly is commonly called a caterpillar, the larva of a beetle is called a grub, and the larva of a fly is known as a maggot. Larvae molt periodically, and when mature they transform to an inactive form known as a pupa. The adult moth, beetle, or fly emerges from the pupa after a period of no feeding and little activity, complete metamorphosis. The length of the life cycle varies greatly with many species of insects. Some develop from egg to adult in a few days or weeks. Many require a year, and a few take 2 or more years to reach maturity. Identification. Proper identification is very important so that you don't need to worry about anything because you'll already know there are solutions. Finding the right one comes from identification first. When the insect is correctly identified, information regarding its life cycle, food preference, habits, and whether it is beneficial or harmful can be looked up anywhere online or specific apps that identify insects. Beneficials. Less than 0.5% of all insects are pests on moringa trees. Many beneficial insects feed upon harmful ones. Common examples of predators are lady beetles, praying mantis, assassin bugs, lacewings, and parasitic wasps. Spiders also prey on numerous insect pests. Many species of mites do not damage moringa trees but instead feed upon other plant-feeding mites. Also, many harmful insects are destroyed by tiny wasp-like parasites. It is important to learn to identify these beneficials and to recognize when they are holding pests in check. There are several types of native ladybugs and Asian lady beetles. These are among Florida's most beneficial insects. Both adults and larvae prey on aphids, scale, mealybugs, mites, and other soft-bodied insect pests. Adults are oval, orange, or reddish with black markings, or black with yellow or red markings. Most species are about 1/4 long, but range from 1/16 to 1/2. The larvae are elongated, with a cone-shaped abdomen somewhat flattened and covered with small spines. They are usually dark or black with brightly colored spots or bands. Some larvae are white and resemble mealybugs. Both adults and larvae are frequently found feeding among aphid populations. Praying mantids are large insects. Adults are usually more than 2 in length. Some are brown and others are green. The front legs are modified for grasping and holding their prey. They wait within the foliage, legs in an upraised position for prey insects. This characteristic has given rise to the common name praying mantis. Nymphs and adults feed on a wide variety of insects, including flies, honeybees, crickets, and moths. They are not effective predators on aphids, mites, or most caterpillars. Assassin bugs are generally black or brown, but many of them are brightly colored. They are 1/2 to 1 in length. The head is elongate with a short, curved beak. These bugs are usually found on the leaves, where they attack many harmful insects. Caterpillars are their favorite prey. Many species will inflict a painful bite if handled. Lacewings are common insects found on grass, weeds, cultivated row crops, shrubs, and trees, but have been spotted on moringa trees. Most adults are greenish with copper-colored eyes and are about 3/4 in length. The adults may be predaceous or may feed on pollen. The white eggs are attached to the leaves by thin stalks, about 1 furlong tall, thus preventing the larvae from preying on each other. The larvae are elongate and large, with sickle-shaped mandibles. They are predaceous and feed primarily on aphids, thus the common name aphid lion. Parasitic wasps are an extremely important group of beneficial insects. These wasps are very small, most are less than 1/2. A parasitic wasp depositing its egg within an aphid is 1/8 long and usually are not noticed. They lay their eggs on or in the body of a host insect, and the immature stage consumes the host's tissues over a period, eventually killing it. Pupation may occur in or on the host. Some wasp larvae construct many small white cocoons on the body of their hosts. Other species pupate inside their host, and the emerging wasp leaves a small circular hole in the host's body as evidence of parasitism. These wasps parasitize many harmful insects such as scales, whiteflies, aphids, leaf miners, and caterpillars. Spiders are predaceous, feeding almost exclusively on a wide variety of insects, paralyzing their prey with venom injected affected by their bites. They rarely bite man, and only a few, such as the widows and brown recluse spiders, are dangerously venomous. The majority capture their prey in webs. However, many of the more beneficial species, such as the wolf, jumping, and crab spiders, do not spin webs. Those that do not construct webs are especially effective in capturing insects that inhabit the soil surface or on moringa tree leaves. Groups of pests. Pests may be divided into groups according to the way they damage plants. These insects have beak-like mouthparts, which are used for piercing the plant tissue and sucking the plant juices. Examples include scales, aphids, whiteflies, mealybugs, thrips, iase bugs, and spittlebugs. Scales are the most serious pests on many moringa tree farms. There are many species of soft and armored scales present in Florida. They vary in size from 1/16 to 1/2 in diameter. Both types damage plants by sucking juices from them. Scale insects attach themselves to moringa trees soon after hatching, and rarely do these insects move from their feeding site during their lives. Scale insects feed by inserting a tiny thread-like beak into many plants, including moringa trees, and sucking the juices. Scales feeding on the undersides of leaves may cause yellow spots to appear on the top sides. These spots become larger as the scales continue to feed. Heavily infested moringa trees appear unhealthy and produce little new growth. If the scales are not controlled, leaves will drop prematurely, sometimes killing portions of twigs and branches. The moringa drumsticks will not fully develop and mature on a tree infested with scales. Scales secrete a wax covering their bodies, making them more difficult to control as they mature. Weekly inspections of plants during the spring, summer, and fall are necessary. Carefully observe the undersides of leaves and stems for scale infestations. Soft scales excrete large amounts of honeydew, which provides an excellent medium for the growth of a black fungus called sooty mold. Armored scales do not secrete honeydew. In some cases, sooty mold may be detected before the scale population if monitoring has not occurred. Many species of scales are highly parasitized by tiny wasps. A small hole in the wax covering indicates parasitism. If 20–40% have been parasitized, continue to monitor the infestation because a pesticide application probably is not necessary. Egg hatching of scale insects is closely correlated with the flush of new growth in the spring. Newly hatched scales or crawlers move about the plant until they locate succulent new growth. They insert their piercing, sucking mouthparts into the plant and begin feeding. If your moringa trees have a history of serious, persistent scale problems, they should be sprayed in the spring, shortly after the new growth hardens and before the scales become mature. The crawler stage is the most ideal time to control scale. Some armored scales reach the adult stage in 6 weeks, and there are several generations per year. Mealybugs are pests of moringa trees, in addition to some other plants and woody ornamentals. They excrete honeydew that attracts ants and serves as a medium for development of sooty mold fungus. Mealybugs are soft-bodied scale insects, which are usually covered with white, powdery, or cottony wax-like materials. Short wax projections extend from the margin of the body, and some species have long filaments projecting from the rear of the body. They vary from 1/8 to 1.5 in length when mature. Mealybugs have piercing, sucking mouth parts, and damage appears as chlorotic spots on the leaves, drumsticks, or seeds. They tend to congregate together, looking like fluffs of cotton on the foliage. The life cycle takes about 30 days at 80 degree F. Aphids, or plant lice, are small, soft-bodied, pear-shaped insects with sucking mouthparts. They may infest almost any plant. Aphids cause damage by sucking plant juices. However, their ability to transmit plant viral diseases may be more harmful than direct feeding damage. Aphids are generally less than 1 to 8 long and vary in color from green to reddish to black. The most distinguishing feature when identifying aphids are the 2 short tubes, or comicals, extending from the rear. Aphids are commonly found on young, developing leaves and stems in clusters or colonies of individuals. Their feeding distorts a plant's new growth, and the leaves curl. Aphids are unlike most insects. Almost all aphids are female, reproduce without mating, and seldom lay eggs, instead giving birth to live young. Aphids can reproduce rapidly and produce many generations in a year. Each female aphid produces 5,100 daughters during her life, and each daughter begins reproducing in 6 to 8 days. Examine plants weekly, especially during the spring months when new growth is abundant. Observe for any predators or evidence of parasitism before applying insecticides. Aphids parasitized by tiny wasps will have a small hole in their back, and their body becomes hard and round. Ladybugs, beetles, and aphid lions are also important predators of these pests. Whiteflies are common pests on moringa trees. Adult whiteflies resemble tiny white moths. However, they are not closely related to moths, but are more related to scale insects. They are only 1/16 long and have 4 wings. The wings and body are covered with a fine white powdery wax. The immature stages, nymphs, found on the undersides of leaves are flat, round, and slightly smaller than a pinhead. They are light green and somewhat transparent. Whiteflies have piercing sucking mouthparts that puncture the leaf and suck the plant juices. Top sides of the leaves on infested plants become pale, chlorotic, or spotted from the insects feeding on the undersides. Whiteflies can transmit viruses among plants. Females deposit eggs on the undersides of the leaves. In 4-12 days, the eggs hatch into active 6-legged nymphs, or crawlers. The crawlers insert their mouthparts into the leaves and remain stationary for the remainder of their immature stages. The life cycle from egg to adult takes about 30 days at 80 degrees F. The silverleaf whitefly received its name due to the characteristic damage it causes on leaves, a silvery appearance to the upper surface of the leaves due to whitefly feeding on the undersurface of the leaves. It is difficult to control chemically because it has the genetic ability to become rapidly resistant to pesticides. Females can produce from about 6,300 eggs, depending on temperature and the host plant they are feeding on. Newly laid eggs are smooth and whitish yellow. As the eggs approach hatching in about 5-7 days, they turn brown. The immature nymphs are thin, flat, elliptical, and greenish yellow. They are sedentary, apart from the crawler, which after hatching usually moves only a very short distance. Once a feeding site is selected, the nymphs do not move. In heavy infestations, they can be so numerous that they almost cover the entire underside of the leaves. Whiteflies have insect enemies. These include adult and immature ladybug and beetles, lacewings, a thrips species, mites, tiny wasps, and ants. Lacebugs are small, broad, flat insects about 1/8 long. Their bodies are usually brown and wings are clear with a fine lace-like appearance. Some species have a broad hood-like projection that extends over the head. Immature lacebugs are black, wingless, and covered with spines. They have piercing, sucking mouthparts, and damage appears on the top side of the leaf as a whitish speckling, which is caused by the insects feeding on the undersides of the leaf. The presence of shiny black spots of excrement on the undersides of leaves is a good indication of a lacebug infestation. In addition to the excrement, there is often a brownish spotting on the leaves. This is a protective coating that many lacebug species secrete over their eggs to protect them. Lacebugs prefer sunny locations. Lacebugs often have very specific host preferences, which aids in identification. Lacebugs have several important natural enemies, including egg parasites, lacewing larvae, assassin bugs, spiders, and predatory mites. Thrips are very small, slender insects with rasping, sucking mouthparts used to remove plant juices. Their 125 to 1/8 body is yellow-brown or black. Thrips' life cycle takes 2 to 4 weeks, with 3 to 5 generations per year. Thrips populations peak during the spring months. Some species, like the greenhouse thrips and red-banded thrips, are primarily foliage feeders. Others, such as Florida flower thrips and gladiolus thrips, feed on flowers. Infested leaves have a stippled appearance, and small brownish specks of excrement usually are visible on the undersides of the leaves. Infested flower buds fail to open, or the flowers are deformed. Damaged flowers become streaked and discolored. Shake flower or leaves suspected of being infested over white sheet of paper to detect the insects. Chewing pests or foliage-feeding insects. Chewing insects may feed on the leaves, flowers, or attack the roots. Examples include caterpillars, beetles, grasshoppers, and katydids. Caterpillars are the immature or larval stage of moths and butterflies. There are numerous species, ranging in size from minute to over 2 long, that feed on moringa leaves. Caterpillars vary in color and may be smooth, hairy, spiny, horned, or possess some other characteristic used to camouflage or protect themselves against predators. They devour the foliage, leaving holes and irregular areas, or they may strip leaves entirely. The extent of the damage depends on the size of the caterpillar. Caterpillars leave excrement as they feed, known as droppings. Cutworms are larvae that typically stay in the soil during the day and feed at night on the base of tender leaves, cutting them off at the soil line. Some climb up the plant and feed on buds and leaves. Grasshoppers and katydids are insects that occasionally consume large quantities of foliage on moringa trees, leaving an ugly, irregular appearance of the leaf. They are common insects in Florida's natural areas. Adult grasshoppers deposit eggs clumped together in pods in the soil. Normally, there is only one complete life cycle per year, but several species have more than one generation. Feeding habits vary greatly. Some species feed only on grasses, some only on broadleaf plants, while others feed on moringa leaves. Many species will consume green and dried moringa sticks and material. Often grasshoppers will disperse when favored food plants are depleted, moving into nearby crops. Grasshoppers are easy to see and should be controlled before they become numerous. Katydids, which are green and feed at night, are not commonly found in large numbers. Beetles are hard-shelled insects with chewing mouthparts. Most beetles are either plant-feeding, predaceous, or scavengers. Beetles have complete metamorphosis with egg, larva, pupa, and adult. Some species have different feeding habits in different stages, such as those with predaceous larvae and plant-feeding adults. But in many species, the adult and larva share the same diet. Beetles range in size from minute to over 6 in length. Adults possess hard, tough, or horny forewings that serve as a protective covering for the delicate hindwings. The forewings are folded over the back and meet in a straight line over the abdomen, but are held stiffly out to the sides when the insect is in flight. Beetles that eat moringa chew off parts of the foliage or flowers. Small ones love to burrow in the drumsticks and seeds. If left on the trees for more than one season, the moringa trees can get infested and spread to new drumsticks. Some beetles are active at night and hide beneath the plant in debris or mulch during the day, while many feed during the day. Flower beetles are difficult to control as they often fly in from adjacent areas in large numbers to feed on petals, nectar, or pollen. Beetle larvae are also destructive. They may feed on the roots or bore through the stems and branches. Some beetles feed on sap that flows from a tree wound. Spider mites are not insects, but are closely related to spiders, ticks, and scorpions. They suck plant juices with their piercing, sucking mouth parts. Spider mites are among the most common pests that attack moringa trees in Florida. Often, the damage is severe before an infestation is realized because these pests are very small and usually go undetected on the undersides of the leaves. Damage from light infestations appears as yellow or gray stippled patterns, small touches or dots of color on the leaves. The undersides of infested leaves usually have fine, silken webbing spun across them. Heavy infestations cause the leaves to turn yellow, gray, or brownish and eventually drop off. Webbing may be spun over entire branches, or in the case of small plants, over the entire plant. When the undersides of the leaves are examined closely with a 10 or 15 power magnifying glass, the small mites can be seen. Mature spider mites have 8 legs and are only 1/50 of an inch in length or smaller. They may be light green with 2 dark spots on the sides of the abdomen, red or purplish. The adult female lays several hundred eggs during her life. The eggs hatch in about 3 days. Immature mites molt 3 times before reaching the adult stage. It is the process of molting, shedding the old skin, that produces the grayish residue seen on the leaves. Under optimum conditions, 80° F, mites complete their development from egg to adult in 7 to 10 days. There are many overlapping generations per year. Spider mites are frequently found on outdoor and interior moringa trees. Syringing indoor plants with water but not saturating them can help control spider mite infestations. Wicking and watering trees from below without getting the foliage or leaves wet. Learn to recognize mites and their injury. Examine plants at weekly intervals and treat them promptly when damage appears. Mite infestations are more severe during hot, dry weather. There are also many species of predatory mites that prey on and kill the plant pest spider mites. Lacewing larvae and some species of thrips, ladybugs, and minute pirate bugs are also effective predators of plant feeding on spider mites. Leaf miners are very small larvae of flies, beetles, or moths that tunnel or mine between the upper and lower leaf surfaces as they feed. New leaves infested with leaf miners are often stunted and deformed. The 2 most common kinds, the serpentine and the blotch leaf miners, are so named because of the shape of their mines in the leaves. Leaf miner eggs are inserted into leaf tissue, where the tiny larvae hatch and make cavities between the upper and lower leaf surfaces. Pupae will be found at the center of a blotch mine, or at the end of gradually enlarging serpentine mines. Both stages are very well protected from natural enemies. Although the leaf miners do not generally move from leaf to leaf, a heavy population of these insects can cause widespread aesthetic damage to the host plant. Infected leaves should be removed and discarded, and pruning may be required in the event of a heavy infestation. Borers are a type of insect that bore into the twigs or trunks of moringa trees. Examples are pine bark beetles, sea grape borer, and carpenter worm that make their way to some moringa trees if the conditions are right, but the goal is to prevent conditions that cause these pests to arrive and stay. While there are many kinds of borers, most are the larvae of beetles or moths. They can be placed into three main types. The pine bark beetles, which bore in the inner bark and feed on the cambium. 2. Borers that burrow in small limbs or twigs. And 3. Borers that burrow deep into the trunk. Usually, sawdust-like borings are noticed around the entrance holes and collect in bark crevices. Sap may flow from holes and form small pitch tubes. In most cases, the borers are not the primary cause of trouble to trees, but rather these insects attack trees first weakened by something else. A tree does not have to be badly weakened to make it susceptible to attack by borers. Injury or stress to the tree caused by drought, saltwater intrusion, soil added or removed above the roots, soil compaction, digging house foundations, septic tanks, underground utilities, lightning, winds, storms, freezes, and wounds to the trunk, or roots caused by vehicles or machinery may mark the beginning of insect problems. Also, the setback that trees receive in transplanting increases the possibility that borers may attack them. Other pests. Sooty mold is a black fungus that grows in the excretion of aphids, mealybugs, many soft scales, and particularly of immature whiteflies. This fungus detracts from the beauty of ornamental plants, but does not cause as much injury as most people believe. Controlling the above pests will prevent or reduce the problem of sooty mold. Ants are fond of honeydew excreted by aphids, mealybugs, and certain scales, and they may protect and move these pests around from plant to plant. They are social insects with biting mouthparts that live in colonies. Ants, in some cases, can inflict painful bites or stings. Many species of ants are predatory, feeding on the larvae or pupae of harmful insects. Some could be helpful, but sometimes ants may injure seedlings, cut leaves, steal seeds, and cover spots of the moringa trees near the base and destroy the soft wood, making their nests and homes in the trunk of moringa trees, especially the area that stays shaded and moist. The red imported fire ant has become a major agricultural and urban pest throughout the southeastern states. Fire ants have an impact on the ornamental plant, sod, and landscaping industries because of problems associated with shipping infested plant material into uninfested areas of the country. Nursery inspections, compliance agreements, and certification are used to contain the spread of red imported fire ants. Slugs and snails are mollusks and are similar in structure, except slugs lack the snail's external spiral shell. They move with a gliding motion by means of a long, flat muscular organ called a foot. This muscle constantly secretes mucus, facilitates movement, and leaves a silver-like slimy trail that signals the presence of either pest. Snails and slugs are most active at night and on cloudy or foggy days. They prefer damp and shady areas. On sunny days, they hide under moringa leaves and dense ground covers out of the heat and bright light. During dry weather, most slugs and snails hibernate hidden under logs or stones or buried in the earth, so it's important to keep your grow area as clean as possible. Snails and slugs feed on decaying plant matter, as well as on a variety of living plants by chewing irregular holes with smooth edges in leaves and flowers. The silvery mucus trails confirm slugs or snails, not earwigs, caterpillars, or other chewing insects, cause the damage. Snails and slugs favor seedlings and moringa trees' succulent foliage. They are more of a serious pest of ripening fruits, such as strawberries, that are close to the ground. They will also feed on foliage and fruit of some trees. Citrus are especially susceptible to damage. Most moringa trees are also not seriously affected, but it is important to identify if you have other fruit trees growing nearby. Slugs have soft, unsegmented gray to black or brown bodies. Their head has a pair of upper tentacles bearing eyes and shorter pair for smelling. The mouth is found at the center of the head with the mucus gland below. Snails have the additional advantage of having their bodies protected by a hard shell, usually marked with spirals. Snails and slugs have many natural enemies, including ground beetles, pathogens, snakes, toads, turtles, and birds, but most are rarely effective enough to provide satisfactory control in the garden. Clean surroundings will aid in controlling slug and snail populations, and removal of boards, sacks, piles of brush, and other debris will limit their numbers. Chapter 6.2 Moringa Diseases. Disease pathology. Diseases continue to have identifiable impacts across the spectrum of agricultural crops. Some may only spread to similar species or root types, while others can be spread from one crop to another given the area of each operation. Wind and weather can play a role in the scale, and speed diseases can spread amongst crops. The southern cornleaf blight epidemic of 1970 cost Florida growers at least $5 million. Blue mold, a long-forgotten disease of tobacco in the plant bed, suddenly became epidemic in the field during 1979 in the USA and Canada. Continued epidemics occurred in 1980 and 1983. Controls for these diseases have been effective but expensive. Many Irish descendants in this country are here because a disease called potato late blight starved their forefathers out of Ireland in the mid-1800s. Soybean stem canker was epidemic in 1983. Virus diseases have caused destruction of watermelon and squash plantings. Tomato spotted wilt virus has been found in Florida in tomatoes, peanuts, tobacco, and gladiolus. The citrus industry in Florida was tasked in the early 1900s and again in the mid-80s and mid-90s by citrus canker, a disease affecting the root system caused by a bacterium. Stringent quarantines and the eradication of millions of citrus trees were used for control of citrus canker. More recently, it has been the greening disease affecting citrus trees, a disease spread by an Asian bug eating the citrus, spreading the bacteria with more noticeability in the leaves. Sudden oak death syndrome, SOD, was identified in 2004 in Florida, potentially arriving from the West Coast. It is a fatal fungal disease affecting a broad range of ornamental species with no known cure. SOD also affects healthy trees in the forest ecosystem, potentially destroying habitat and food supply for a wide variety of wildlife. Quarantines and eradication programs, in conjunction with extensive surveys, have proven the most effective way to deal with infected plant material. Other diseases that are typically less newsworthy than the above-mentioned diseases occur commonly. Diseases such as peanut leaf spot, bacterial spot of tomatoes and peppers, black rot of cabbage, pythium root rots of turf grasses, and rhizoctonia stem rots are but a few diseases that occur each year in Florida. Control of these diseases is achievable when growers use production techniques that are proven to be effective and within the economical limitations of the crop. The American chestnut tree, for practical purposes, has been eliminated from this country because of a fungus disease called chestnut blight. The dreaded Dutch elm disease continues to scourge the country, and lethal yellowing of coconut palms has changed the tree line of South Florida. Control of plant diseases requires extensive efforts in some situations and minor adjustments in other situations. The science of disease pathology is charged with the opportunity to reduce the impact on moringa diseases. Since we plant moringa trees in polycrop systems, with all these diseases potentially near moringa trees, successful curtailment of other plant diseases will be a crucial skill to obtain for the longevity and health of your food forest. Moringa disease. A moringa tree becomes diseased when its chemistry or structure has been submitted to an abnormal, sustained alteration. This definition, although vague, is usable. It tells us that if, for example, a leaf is pulled off a tree by a person, that is not a disease, but instead an injury because the alteration is not continuous. A fungus, bacterium, or virus, on the other hand, enters the moringa tree and continues to deprive it of nourishment or continuously alters normal functions. The lack of a nutrient over a period likewise causes a continuous dysfunction resulting in undesirable symptoms. Moringa diseases are caused by either non-living or living agents. Non-living agents include low temperatures, high temperatures, atmospheric impurities, mineral deficiencies, mineral excesses, or possibly other causes. The living agents that cause moringa diseases are fungi, bacteria, a few higher plants, nematodes, algae, viruses, mycoplasmas, and viroids. Fungi are plants that lack chlorophyll, stems, leaves, and roots. Their vegetative body is made up of microscopic tubular structures called hyphae. Fungi are further characterized by the production of microscopic seeds called spores. A spore of fungus is approximately 1/250 of an inch long. Fungi produce different types of spores. Some spores are suitable for wind or water dissemination, while others have thick walls, thereby being adapted for survival in soil or other concealed places for many years. Some spores serve as carriers of genetic traits for sexual reproduction. Many fungi can live as saprophytes in the soil or decaying plant litter, as well as being parasitic. Fungi that can grow saprophytically on old moringa material debris and soil usually can be grown as a culture on a growth medium in the laboratory. Some fungi, however, such as rusts, downy mildews, and powdery mildews, are obligate parasites. That is, they normally grow only in a plant. Some fungi produce microscopic or macroscopic modified hyphal aggregates called sclerotia, which serve as survival structures. Bacteria are microscopic, one-celled organisms that increase by division of cells. Some bacteria can divide every 20 minutes. In 24 hours, this could result in 300 billion new individuals. A typical bacterium is 1/12,500 of an inch long. Bacteria can be grown as cultures in a laboratory. Bacteria survive on or in host plants, susceptible weeds, and organic debris in soil. Within the last 15 years, a group of bacteria that are limited to xylem tissues of plants were identified as causing moringa diseases. They tend to be smaller than other bacteria-causing diseases, and some are culturable in the laboratory. They are transmitted by leafhoppers and can be transmitted through grafting as well. Using those same blades to harvest moringa trees without sterilizing could spread the bacteria from other plants to your moringa trees. Viruses are typically particles made up of a nucleic acid core and a protein coat. No cellular structure is present, although some may have been closed by a membrane. Viruses reproduce only in living cells of susceptible host plants. Virus particles are not visible with light microscopes. An electron microscope is used to reveal their structure. A typical virus particle is 1/80,000 of an inch long. Viruses are obligate parasites because they grow only in living plants. Viroids are similar to a virus, but lack a protein encapsulation. Viroids causing plant diseases contain RNA only, and, therefore, are the smallest known infectious agents causing plant diseases. Avocado sunblotch, Citrus exocordis, and chrysanthemum stunt are either caused by or associated with viroids. More research is taking place now to know the effect viroids have with moringa trees specifically. Algae resemble fungi in size and structure, but differ primarily by the presence of chlorophyll in algae and the absence of chlorophyll in fungi. Algae have unicellular, colonial, and filamentous species. A few are parasitic in plants grown in subtropical or tropical environments. Tropical fruit trees such as mango, avocado, and citrus are infected by red algae. If your operation has an irrigation issue and algae forms in the water or on a tree because of a deformed nozzle, it could affect the production of your moringa tree. Mycoplasma have a cellular-like structure, elementary body without a true cell wall. They contain protein, DNA, RNA, and enzymes. The elementary bodies vary in shape and size. Some are 1/2500 of an inch long, and others are 1/10 that size. Also, mycoplasma are sensitive to heat and some antibiotics. Many plant diseases, previously thought to be caused by viruses, transmitted by leafhoppers and in the yellows group, are now known to be caused by mycoplasmas, not viruses. Pathogen dispersal. Pathogens are living agents, usually microorganisms, that cause disease. Fungi have hyphae, which grow on or in the soil, or within or on host tissue. Fungi also have spores, which may be spread by wind, insects, rain, or irrigation water. Some spores have flagella, tails, which allow them to swim in irrigation ponds, rainwater, dew, or soil moisture. Fungi also spread when infected plants, including seed, are moved from one location to another. Similarly, fungi are carried on equipment or maintenance implements or people working within the planting. Bacteria are spread by insects, irrigation water, rain, movement of infected plants, seeds, seed pieces, grafting, and equipment. Viruses are spread by mechanical rubbing of one infected plant on another, insects, fungi, nematodes, transporting of infected moringa trees from one location to another, seeds, seed pieces, grafting, daughter, equipment, and people's hands. Each virus may be spread by certain methods. For example, many legume viruses are spread by seed and aphids, but not by fungi or nematodes. Moringa is not a legume, but it does have high levels of nitrogen. If you were to chop and drop moringa, it would act like a nitrogen fixer, like a legume. Mycoplasma are dispersed by leafhoppers or moving infected plants. Viroids are spread by implements, pruning tools, or other mechanical devices. Pathogen entry. Fungus spores germinate, form a germ tube, and then an infection peg is formed from the germ tube that penetrates directly through the cuticle or stomata of a leaf. Infection of roots and stems occurs either by germ tubes formed from spores or hyphae that penetrate the tissue. Fungi can also infect plant parts when wounds are made by harvesting, equipment implements, hail, wind, blowing sand, insects, nematodes, or other fungi. Bacteria enter plants through wounds or natural plant openings, such as stomata, lenticels, or hydathodes. When plant tissue is gorged with water, bacterial ingress into plant tissue increases. Viruses can enter a plant through wounds, especially during the harvesting and pruning of your moringa trees. A recommended approach is keeping your harvest less than 30% of the entire tree at one time, and to cover the cuts with a liquid plant sealant that reduces infection. Also, aphids transmit many of the viruses that occur in Florida. Other insect types are known to transmit viruses. Some of the wilt viruses is vectored by thrips. When an insect feeds on an infected plant or tree, the virus could pass from the insect into the moringa tree, or the insect acquires the virus from the moringa tree, spreading it to other moringa trees or even other plants. Some nematodes transmit viruses by piercing the root. This can result in passing the virus particle from the nematode into the moringa tree. Corky ring spot virus in potato is transmitted by certain nematodes. Moringa tree roots can resemble a starchy potato-like substance, like cassava. Fungi are vectors for certain viruses. Soil-borne wheat mosaic virus is transmitted by a soil-inhabiting root fungus. Keep your moringa trees high and dry to avoid contamination of any pathogen. Components of moringa disease. Any moringa tree can be susceptible to an agent causing a disease and a suitable environment. Then a disease can occur. For example, fungi that cause leaf spots need a susceptible host, moist conditions on the leaves, and favorable temperatures so that spores will germinate. Many root-rotting fungi need a susceptible host coupled with high soil moisture, or a soil pH favorable for fungus growth. Disease is caused by the agent impairing necessary functions of the moringa tree. Some diseases block water-conducting vessels in the moringa tree, which results in a wilted condition like drought. Root rots destroy the roots that absorb water and nutrients from the soil. Leaf spotting diseases reduce photosynthesis in the moringa tree, which results in less food manufactured by the tree for itself. Seeds, seed pieces, fruits, and flowers may be destroyed by rots or blights. Diseases of this type reduce the reproductive ability of a moringa tree to produce healthy leaves, drumsticks, and seeds. Diseases in the urban environment. Several generalities can be made concerning more commonly encountered diseases in Florida. Many of these diseases require similar environmental conditions to commence, as well as increase in severity. chemical control measures heavily relied upon in past years as the total disease control program are now being integrated with such non-chemical controls as host resistance and cultural practices to manage disease problems. Not all diseases, viruses, bacteria, or fungi affect moringa trees. Much of this research is still being conducted as it's being grown more in the USA. Points to remember about diseases include 1. Not every spot or discoloration or insect is automatically a disease problem. Many of the nondescript disease symptoms of root and crown rot diseases, melting out, etc., can be mimicked by improper cultural practices, misused pesticides, nematode, or insect injury. Remember, diseases are continuous conditions that do not stay static, but increase and spread. It is often faster to rule out the involvement of other factors than to verify the presence of disease. 2. Suspected disease problems cannot always be reliably diagnosed by casual observations. Root and crown rot phases of many diseases require laboratory diagnosis. 3. Diseases can be expected to occur more frequently on poorly maintained moringa trees. Imbalances in nitrogen, phosphorus, and potassium levels, as well as accumulation of sticks and rotting wood on the tree, will result in less vigorous and more susceptible moringa tree diseases. 4. Periods of high humidity, rain, heavy dews, or fog favor the incidence of most fungal diseases of moringa trees. Moisture is needed on the surface for infection to take place. Irrigate early in the day to minimize the period in which the leaves, soil, and roots remain moist. Water deeply when needed. Moringa trees that are wet going into the night period, moist from irrigation, is more likely to become diseased. 5. Most leaf disease problems can be accurately diagnosed from the symptoms expressed by the affected moringa tree. Many of the root and crown diseases are difficult to accurately diagnose due to the frequent interactions of these fungal pathogens with each other, nematodes, insects, or cultural problems. Laboratory diagnosis is essential for these conditions to choose the proper control measures. When laboratory diagnosis becomes necessary, A. Sample the affected areas of the moringa tree prior to fungicide application. B. Take the sample from areas that look susceptible to damage, but aren't damaged yet, close to the site of the concerned area. Grab that area between affected and healthy areas to sample. C. Cut a sample of the moringa trunk or branch from each symptomatically distinct area. D. Never add water to the area or sample, nor seal them in plastic or aluminum foil for transport. E. Deliver sample to your nearest Florida Extension Plant Disease Clinic, FEPDC, or other local diagnostic laboratory. More detailed information about where to go for help in diagnosing plant problems is included in the bibliography. Common diseases in Florida. Except for one disease caused by a virus, mostly all diseases in Florida are caused by fungi. Examples of fungi in your daily life include the molds in the bathroom or refrigerator, the green stuff on an old orange or bread, and the mushrooms you eat. Unlike plants, fungi are unable to produce their own energy and must rely on living or dead hosts for energy and growth. Most fungi living in the soil environment are totally harmless, using only dead organic matter for growth. A very limited number of fungi at some point in their life cycle cause plant diseases by infecting living moringa trees. It is important to know that when a fungal pathogen is not actively attacking the plant, it has not disappeared from the area. It is simply surviving in the environment in a state of dormancy like a bear in hibernation, or as a saprophyte, non-pathogenic phase, living off dead organic matter in the dead soil layers or rotting tree material. There are two common patterns of moringa tree disease symptoms. One is a circular patch on the leaves or bark, either small or large, that is no longer uniformly the same color as the material, leaf or bark. The second is spots on the leaves. For disease patches, examine the leaves and roots in these patches for characteristic symptoms of a disease and signs, actual fungal structures of the pathogen. Leaf spots. 5.1 Brown leaf spot. Symptoms. The symptoms appear to be scattered brown spots on the leaves and then eventually spreading to cover them entirely. Coalescing of spots leads to irregular and blighted appearance of the leaves. The leaves turn yellow and fall off prematurely. Pathogen Cercospora spp is responsible for the disease. The fungus is characterized by dark brown stromata from which arise fasciculate straight or slightly curved, continuous simple yellowish-brown conidiophores producing polymorphous, cylindrical, fusoid, 1-3 septate, yellow to brown, conidia tapering towards the apex. Mode of spread. Conidia are disseminated by wind and rain splashes to the leaf surface where they germinate and cause infection. The spots are produced on the leaves on which conidia are formed, serving as source of secondary infection. When the weather becomes unfavorable, the fungus survives in the infected plant debris and the cycle is repeated next year. Epidemiology. Disease development is favored by high humidity and temperature. The pathogens sporulate abundantly at 20 to 30 OC. Management. Preventative measures, as stated in the previous chapters, organic foliar sprays, 9.2 Septoria leaf spot. Symptoms: circular spots with whitish gray center and dark brown margins are seen on the leaves, stem, and calyx. Center of the spots shows minute black glistening pinhead-sized pycnidia. Severe infection causes defoliation during rainy season. Pathogen. The disease incited Septoria lycopersicon. The young mycelium is hyaline thin, walled, and sparingly septate. Older mycelium is brown, infrequently branched, and septate. In the initial stage of pycnile formation, several hyphae aggregate at certain point, and after intertwining, form a mass of hyphal tissues that assumes the appearance of pseudoparenchyma. Pycnidia are subglobose, composed of 2-3 layers of brown cells. Pycniospores are filiform, slightly curved hyaline, and septate with pointed or rounded ends. Mode of spread. The pathogen overwinters in the infected plant debris in the form of mycelium or spores, or in the debris of solanaceous weed hosts. The conidia are splashed by rains, causing infection. Epidemiology. The fungus has wide host range, including solanaceous crops and weeds growing in and around a moringa field. Temperature of 20 to 25 OC with 75 to 92% relative humidity is congenial for disease development. Management. Preventative measures as stated in the previous chapters. Spray applications of various organic fungicides. 5.3 Alternaria leaf spot symptoms. The symptoms appear on leaves as circular to angular, dark brown spots with concentric circles. The spots coalesce and cause drying and defoliation of leaves. Black or brown marks appear on the branches. Pathogen. Alternaria solani. The mycelium is septate branched light brown hyphae, which become darker with age. Conidiophores emerge through the stomata from the dead centers of the spot. Conidia are beaked. Muri form dark and borne singly, or in chains of 2. 5 to 10 transverse septa and a few longitudinal septa are present in each conidia. Mode of spread. Primary infection takes place through conidia on crop debris in the soil. The secondary spread of the disease occurs through conidia developed on primary spots. These conidia are blown by wind, water, and insects to the neighboring leaves or plants. Epidemiology. The disease is favored by high soil moisture, relative humidity, dew, and rainfall. The optimum temperature ranges between 28°C to 30°C. Management. The onset of the disease is hard to detect. Once the spots have appeared, it is often too late to treat, and defoliation is inevitable. Clearing of weeds, which are often hosts to diseases, should be practiced. Leaves and young shoots should be checked regularly for symptoms of fungal attacks. Neem seed extract can be sprayed on the plants to control the disease. Pięć powdery mildew. Symptoms. The symptoms appear as white to light gray powdery growth on the undersurface of the leaves, and the corresponding upper surface exhibits yellow lesions with brown necrotic centers. Affected leaves curl upwards and shed profusely. Affected plants lose their vigor and remain barren. Yield is very much reduced. Pathogen. The fungus Levolula turica is responsible for this disease. It is an obligate parasite having endophytic mycelium. Conidiophore are long and multi-branched. Conidia are pyriform and cylindrical, borne singly or in short chains. Epidemiology. Temperatures of 25 to 30°C and cloudy weather are highly favorable for the disease development. Management. Spraying wettable sulfur 0.2% gives better control. 5.5 Diplodia root rot. Symptoms. The symptoms of the disease appear as oozing of gum from the affected bark, which remains as an encrustation on the bark. Droplets of gum trickles down the stem. In the later stages, the bark cracks and shreds longitudinally. Leaves turn pale and yellow. Fruit yield is greatly reduced. Pathogen. The fungus inciting the disease is Diplodia spp. Mode of spread. The pathogen spreads through pycnospores with the help of wind and rain splashes. Epidemiology. Reduced tree vigor, insect damage, poor nutrition to the trees, waterlogging, and old age factors. Management. The trees should be kept in a vigorous growing condition. Broken limbs should be cut properly and wound in the bark, especially on limbs, and forks should be scraped and protected with Bordeaux mixture. 6. Fusarium wilt. Symptoms. The affected plants exhibit yellowing of lower leaves in initial stages, and discoloration of younger leaves soon follows. The leaves drop, wilt, and dry. The disease affects a few branches in the moringa tree, or the entire plant may wilt irreversibly. The affected plants or branches dry up. The vascular bundles become brown. Moringa trees are usually stunted in growth, and drumsticks ripen prematurely. Pathogen. The inciting agent of the disease is Fusarium oxysporum, FSP moringae. The mycelium is septate. Microconidia, macroconidia, and chlamydospores are produced. Mode of spread. The fungus is soil-borne and survives in the soil as chromophores, or as saprophytically growing mycelium in infected crops debris. Wind-borne spores, surface drainage water, and agricultural implements also help in distribution of the pathogen from field to field. Epidemiology. Disease development is favored by alternating high and low soil temperatures and high humidity levels. Other favorable factors include light, sandy soils, low soil moisture level, and pH. The presence of nematodes greatly increase infection. Management. Seeds should be planted in disease and nematode-free soil. Seed treatment with carbendazim 2-GKG reduces incidence of the disease. The infected plants should be removed and destroyed. Long crop rotation with non-host crops helps in reduction of the inoculum. Incidence of wilt has been significantly reduced with the application of organic fungicides. 5.7 Fruit rot. Symptoms. Pods reaching maturity showed extensive rot. The disease symptoms are observed all over the surface of the pods, more conspicuously at the stigmatic end. On green pods, elliptical or elongated sunken spots with reddish-brown raised margins can be observed. The pods appear shrunken with thinner dimensions at their stigmatic ends than healthy ones. In advanced stages of the disease, the pods rot and dried up prematurely, leaving uneven raised spots over them. Mode of spread. The causal organism was identified as a fungus Drexlera hawaiiensis. Management. Spraying organic fungicides is effective. Other problems. Rust diseases commonly occur in mild to warm, humid weather. Infections appear first as light-colored flecks on leaves. These flecks enlarge into red to orange linear pustules, often along veins. Pustules will first fill with orange spores and later produce brown to black spores. Severe infections will cause moringa trees to thin and appear chlorotic. Slime mold is not a true disease. However, warm, moist weather and periods with prolonged dews will encourage slime mold problems. A soot-like crusted fruiting structures occur on blades of turf. Color may vary from gray to orange to purple. Brown. Fungi use moringa trees as support structures upon which to reproduce. Slime molds may occur on many species of plants. Take-all root rot. Periods of disease severity coincide with summer and fall seasons in Florida when temperature is high and moisture ample. Stresses contribute to disease development and may include low soil moisture, scalping, nematode injury, soil insect injury. Initial disease development appears as chlorotic patches. Affected areas thin and decline in response to root decaf because the root fungus decreases water uptake to the canopy and photosynthate movement down to the roots. Random green or chlorotic fascicles will persist in diseased patches of leaves or bark. All cultivars of moringa trees are susceptible, especially when grown under stressful conditions. Chapter 6.3 Nematodes. Parasitic nematodes. Moringa trees can get attacked by nematodes too. There are so many microscopic pathogens that can disrupt the living organisms in the soil in and around moringa trees. In this case, we will cover nematodes that attack moringa trees and live in the soil. An unbelievable war that seems to be waged without retreat. All crops grown in Florida are susceptible to nematodes, causing farmers millions of dollars in crop loss annually. Nematodes also are of great concern to the homeowner since they cause severe damage to other plants like turf grasses, ornamentals, and home gardens. Moringa trees are no different. We are often unaware of losses caused by nematodes since they are hidden from sight, and much of the damage caused by them goes unreported or is attributed to other causes. Morphology and anatomy. Nematodes that attack moringa trees are microscopic worms, ranging from 0.25 mm to 3.0 mm, 1/100, 1/8 inch long. They are generally cylindrical in shape, tapering toward the head and tail. Females of a few species lose their worm shape as they mature. becoming greatly enlarged in diameter and assuming varying forms such as pear, lemon, or kidney shapes. Despite their small size, nematodes are complex in organization. Parasitic nematodes possess all the major organ systems of higher animals except respiratory and circulatory systems. The latest discovery shows a multi-layered cuticle which bears surface marks that are used when identifying nematode species. The cuticle of most is transparent, so that sufficient anatomical detail can be seen with the aid of a low-power dissecting microscope. Life cycle and reproduction. Parasitic nematodes have a simple life cycle with 6 stages: egg, 4 juvenile stages, and adult. Male and female nematodes occur in most species, and both may be required for reproduction. However, reproduction without males is common, and some species are hermaphroditic. Females produce both sperm and eggs. Egg production by the individual completes the cycle. The length of the life cycle varies considerably depending on nematode species, its host plant, and the temperature of its habitat. During summer months, when soil temperatures are in the 80s, many nematodes near moringa trees complete their life cycles in about 30 days or less. The number of eggs deposited by a female varies among species and is affected by their habitat. Most species produce between 50 and 500 eggs, but a few occasionally produce several thousand eggs per female. Nematodes and moringa trees. Nearly everyone who grows anything in their yard or garden or landscape in Florida eventually has encountered nematodes unknowingly. They damage so many kinds of plants that many people just throw their hands in the air and give up more often. Is there any way to escape nematode damage in the garden, annual bed, and permanent landscape? Moringa trees that have exposed soil sand have different problems than when grass is growing up to the trunk. Both accumulate pest problems. If there are serious nematode infestations in the roots, soil ball, or planting site, the chances of satisfactory growth are slim, and the longevity of the tree may be substantially reduced. Many moringa trees planted in raised beds for intensive planting are similarly at risk. If planted in soil heavily infested with injurious nematodes, they develop serious root problems, grow poorly, and often fail much sooner than they should. There presently are no cure treatments, but the kind of start and care given either orchard or intensive methods can have a substantial effect on its performance. Reference: the information which follows is adapted in part from Nematology Plant Protection Pointer 21 Nematodes of Landscape Plants and NPPP 35 Annual Ornamentals Nematode Management. Symptoms of nematode injury. Above-ground symptoms of nematode injury are like those resulting from many kinds of root damage. Moringa leaves will lose their luster and wilt more easily than they should. Prolonged stress because of nematode injury to the root system may result in yellowing and eventual loss of foliage. New flushes of growth are weak, with fewer and smaller leaves than healthy plants. Once the moringa trees are woody, they have a better chance of survival if they've been exposed to nematodes. They may suffer the loss of smaller green growth and new stems if the stress continues. If undesirable conditions persist despite use of extra vitamins and minerals, biostimulant, and adjusting pH of the water and adding organic matter to the surface may be needed. You may notice symptoms in the plants closest to the affected moringa tree, but not on all plants or trees. The distribution pattern of damage is usually irregular since nematodes do not typically spread evenly in the soil. Each moringa tree root looks different, so the nematodes may find some root systems more desirable to live and procreate. Symptoms will vary widely from tree to tree. Some kinds of nematodes cause tissues on which they feed to grow strangely, i.e. root knot, and some foliar nematodes. Some stop the growth of the roots, others kill the cells on which they feed, leaving patches of dead tissue as they move on. Fungi and bacteria, which cause root rots, wilt, and other moringa diseases, often infect nematode, damage roots earlier and more severely than uninjured roots. Therefore, depending on the kinds of nematodes involved, nematode damage to moringa trees may include galls, stunting, and decay of roots. Damaged roots are often darker in color than healthy roots. Among the dozens of nematode species associated with moringa trees, relatively few seem to cause most of the serious problems. The root knot nematodes are by far the most noticeable in Florida. Their easily recognized galls on roots make their presence obvious. Galls result from growth of plant tissues around juvenile nematodes that feed near the center of the root. Root knot gall tissue is firm without a hollow center and is an integral part of the root. Removing a root knot gall from a root tears root cortical tissue. Do not confuse nodules formed on roots of many legumes because of beneficial Rhizobium spp. Nitrogen-fixing bacteria with root knot galls. Most natural nodules or bumps are loosely attached to the roots and have hollow centers. Active Rhizobium nodules have a milky fluid in their centers. Moringa trees do not normally have knobs or nodules like nitrogen fixers because moringa does not fix nitrogen in the soil. It does contain high amounts of nitrogen and can be used as a living mulch or chop and drop to add nitrogen to the soil that way. Not only are their symptoms the most easily recognized, but root knot nematodes cause greater damage to moringa trees than other nematodes. Since moringa has wood on it, and that is the most popular food trait for the root knot nematodes, moringa trees can be susceptible to root knot nematodes. We do not know why these unusual infections happen, but it is likely that some moringa trees get stressed from environmental factors or other pests may promote them. Reduce the effects from root knot nematodes with healthier soils. The less distinct discoloration, stunting, and decay of roots that are caused by nematodes other than root knot are easily masked by or confused with the effects of numerous pathogens and other root problems. Nematodes such as lesion, burrowing, and reniform probably cause far more damage to moringa trees than is recognized because their effects are not as readily recognized as those of root knot nematodes. Susceptibility to one kind of nematode is not necessarily related to the reaction of the same nematode on another moringa tree. Nematode assays are common laboratory tools used to identify nematode species. Laboratory soil sample analysis is the only way to determine all the kinds of nematodes associated with a problem and may be necessary to identify the most effective control measures. A nematode sample kit for sending samples to the Florida Nematode Assay Laboratory is available from Florida Cooperative Extension Service county offices. It contains packaging materials, a sample information form, and instructions for collecting and submitting good diagnostic samples from many kinds of crops, plants. Diagnosis of nematode damage. Since nematode populations are distributed erratically in soil, great clusters in numbers within a few feet is common. Above-ground symptoms also appear in areas of irregular size and shape. Nematode damage rarely ends abruptly. If an area of stress is distinct with sharp boundaries where the moringa trees are good and abruptly meet with poor conditions, the problem is most likely caused by something other than nematodes. Roots injured by nematodes are usually dark and short, with few lateral or feeder roots. Root tips may be swollen, and there is often excessive root rot. The most common symptom is a very short root system that does not hold soil together, as well as a healthy root system does when the roots are exposed around the moringa tree in question. The previous history of an area can help determine the likelihood that nematodes are causing a current problem. If diagnostic samples were taken or specific pest problems identified earlier, those records may suggest the pests most likely to be present in the area on this occasion. Especially helpful are records of previous diagnoses from nematode assay, plant disease, and insect identification laboratories. Check with the previous owner or county records to find any clues of past nematode issues. Soil samples can be key diagnostic aids. In Florida, nematode sample kits, which contain instructions and packaging materials for taking and submitting samples to the Florida Nematode Assay Laboratory, can be obtained at county extension offices. There is a charge for processing the samples. Other laboratories also provide nematode assay service in some areas of Florida. Consult with their representatives for prices and services offered. Since most nematode control treatments are expensive, adequate nematode samples should be processed to establish the need for treatment before it is initiated. Chapter 6.4 Weeds. In any discussion of weeds, there must first be a definition of a weed. Many definitions have been used. For the purposes of this manual, a weed is defined as any plant that is a hazard, nuisance, or causes injury to moringa trees. It should be obvious that almost any plant can be a weed under certain circumstances. Whether you live in a rural or urban environment, weeds either directly or indirectly influence your everyday life. Weeds reduce yields, quality, and interfere with efficient harvest. These reductions are eventually passed to the consumer, either in increased prices or in poor quality moringa tree products. Weeds interfere with the consumption of water and irrigation. Some weeds can cause physical damage to people if growing in and amongst moringa trees, causing discomfort and possibly increased medical expenses. It is estimated that the current total losses to moringa trees grown in the USA, Florida agriculture due to weeds are approximately in the millions dollars per year. It is also estimated that the current use of herbicides and pesticides to control weeds is more common than natural permaculture and regenerative practices. Since weed control consists of more than using herbicides, this section addresses other methods of weed control and possibly how they may be combined with chemical weed control for greater efficiency and effectiveness. Some locations may not need chemical implementation, but it is a practical method of choice for farmers around the world because of excess cost associated with weeds. Classification of weeds. Weeds may be classified as grasses, broadleaves, and sedges. They may further be classified by the length of their life cycle, annual, biennial, and perennial. The classification of weeds based on the length of their life cycle may not be obvious on visual inspection, but will have a great impact on the selection and/or success of control procedures. Grass weeds. True grass weeds have hollow, rounded stems in their first year, and the next calendar year they'll have joints that are more closed and harder. The leaf blades of many grass weeds have parallel veins, are much longer than they are wide, and alternate on each side of the stems. Some examples are crabgrass, goosegrass, crowfoot grass, sandbur, annual bluegrass, torpedo grass, and vasey grass. Sedges. Sedges are an important group of grass-like weeds. However, they are not true grasses. Sedges are characterized by a solid triangular-shaped stem with leaves extending in 3 directions. There are annual sedges, often called watergrass, and the predominant and difficult to control perennial sedges. Of the latter group, yellow nutsedge is yellowish-green and reproduces by seed, rhizomes, and tubers. The rhizomes radiate from the plant with a single bulb or tuber at the end, which may produce new plants. Purple nutsedge is usually smaller in growth habit than yellow nutsedge, has reddish-purple seed heads, and produces a series of bulbs on radiating rhizomes. Broadleaf weeds. Broadleaf weeds are a highly variable group of plants, but most have showy flowers and net-like veins in leaves of varying shapes and sizes. In general, they are easy to separate from grasses due to their different leaf structure and habits of growth. Some examples of broadleaf weeds are cudweed, creeping charlie, henbit spurges, burning nettle, pennywort, creeping beggarweed, cocklebur, sicklepod, and Florida beggarweed. Annual weeds. Annual weeds, as the name implies, complete their life cycle within 1 year. They germinate from seed, grow, mature, produce seed, and die in 12 months or less. They may be annual grasses, sedges, or broadleaved weeds. Their life cycle may begin at different seasons of the year. Thus, summer annuals emerge in the spring, mature, produce seed, and die before winter each year. Weeds such as crabgrass and cocklebur are typical summer annuals. Similarly, winter annual weeds sprout from seed in the fall and complete their life cycle before summer of bluegrass, and chickweed are examples of winter annual weeds. Biennial weeds. Compared to annual weeds, biennial weeds are few. These weeds have a 2-year life cycle. They germinate from seed in the fall, develop large root systems and a compact cluster of leaves during the first year. The second year they mature, produce seed, and die. Examples of biennial weeds are cudweed, Carolina false dandelion, wild carrot, and bull thistle. Perennial weeds. Weeds that live more than 2 years are perennials. They reproduce by vegetative parts such as tubers, bulbs, rhizomes, underground stems, or stolons, above-ground stems. Some also produce seed in addition to vegetative reproduction. During the winter season, most survive in a dormant state, and many lose their above-ground foliage and stems. With the beginning of spring, they regenerate food reserves in their root systems. Torpedo grass, nutsedge, and various vines are members of this group of weeds. Florida betony is a perennial weed that, under a subtropical climate, initiates its growth in the fall, grows during the winter months, and goes dormant during the heat of summer. Perennial weeds may be further divided into groups based on the type of root system and reproductive process. Simple perennials reproduce by seeds, but root pieces disturbed by cultivation or other mechanical means will produce new plants. Florida betony is characteristic of this group. Bulbous perennials reproduce by seed and above or below ground bulbs. Yellow nutsedge and wild onions have their bulbs below ground, while wild garlic has an above ground bulb. Creeping perennials, which produce seed and produce rhizomes or stolons. Bermuda grass, torpedo grass, and purple nutsedge produce these specialized stems, rhizomes, and stolons that act as food storage organs and can initiate growth at each node joint along the stem. Obviously, the perennial weeds are the most difficult to control because of their great reproductive potential and persistence. In annual cultivated row crops, annual weeds such as crabgrass, goosegrass, cocklebur, and ragweed are the most prevalent. However, perennial weeds such as nutsedge, Johnson grass, and common Bermuda grass may also be severe problems. In pastures and rangeland, perennial weeds tend to dominate among the perennial forage crops. Weeds such as smut grass, dog fennel, wax myrtle, and prickly pear are very competitive but would not survive in a cultivated row crop situation. Similarly, in moringa tree orchards, where they also perennials in Florida, weeds such as guinea grass, vasi grass, and milkweed vine are perennial problems. Chapter 6.5 Pest Management. Pest Management Control, PMC, is often most effective when used as a component of an integrated pest management system. PMC is an approach to managing insects, mites, pathogens, nematodes, weeds, and other pests by combining a variety of strategies and pest management tactics. PMC utilizes the most appropriate cultural, biological, and non-chemical strategies for managing pests in and around your moringa trees. Chemicals or synthetic pesticides are not organic or recommended at any time near food crops like moringa trees. Plant inspection. PMC can be viewed as a series of steps that is repeated or modified as needed. Prevention, avoidance, monitoring, decision-making, intervention, evaluation. PMC can include the most responsible way to use pesticides if absolutely needed after all other options have been exhausted in careful coordination with other pest management practices. Restricted labeling of pesticides, pest resistance, human safety, and environmental issues are all concerns to nursery and landscape managers. Thus, horticulturists should seek methods of pest control other than scheduled preventative nursery-wide pesticide applications. Integrated means combining a variety of strategies and pest management tactics that can either prevent pest introduction, eradicate pests, or reduce pest populations to lessen their economic impact while maintaining the quality of your moringa trees. Doing nothing is often the most cost-effective option. Some damage can be tolerated, letting nature take its course. Emphasis should be placed on preventative practices such as sanitation, e.g., weed control, removal of plant debris, proper fertilization, irrigation, pruning, etc. Examples of management activities that affect pests and diseases include selection of resistant plant species, regulation of irrigation timing and frequency, alteration of relative humidity levels in the greenhouse, use of least toxic pesticides as needed to preserve beneficial organisms and reduce environmental contamination, or application of biological control agents. Moringa trees should be examined weekly during all times of the year. The undersides of a few leaves on each moringa tree should be checked and the stems inspected for pests. The use of a 10 to 15 power hand lens or magnifying glass aids in detecting and identifying insects and related organisms. Many insects merely rest on the moringa tree and are neither pests nor beneficial. Learn to determine when a pest is present in damaging numbers and to evaluate the potential of the predator or parasite population. To aid in locating small insects such as thrips or mites, a sheet of white paper or cloth may be held beneath the leaves and the foliage struck sharply. The insects or mites will fall onto the paper and can be more easily observed and identified than on the green foliage. Most moringa trees in the urban landscape can be affected by other people spraying, resulting in unnecessary environmental contamination and often upsetting the natural predator-parasite pest balance. Don't apply a control measure until a pest population is present and damage is beginning to occur and you've checked for the presence of natural enemies. Biological controls. Pests in their native areas are usually regulated by predators and parasites that help keep populations at a constant level. Problems occur when pests, but not their natural enemies, are introduced into new areas, and the pest populations increase unchecked. Biological pest control involves use of natural enemies to reduce pest populations, native and non-native, to aesthetically acceptable levels. One inherent principle concerning classic biological pest control, where the agent is introduced only once or on a limited basis for permanent establishment, is accepting that a minimum level of the target pest will always be present. This low pest population is necessary for the biological control agent to have a continual food source after the target pest has been reduced to an acceptable level. Thus, complete elimination of the pest is not feasible when integrating biological control measures into the overall pest management scheme. Homeowners and clients must be educated to this fact and be willing to accept minor levels of pest pressure. Inspection should include scouting for beneficials as well as harmful pests. If such pests as aphids, scales, or whitefly nymphs have a small hole as if stuck by a straight pin, it is evidence that they have been parasitized by tiny wasps. If predators are present or the pests show signs of parasitism, every effort should be made to preserve the beneficial insects. Delay pesticide application and allow the beneficial bugs to control the pest population, or at least choose a method of control or time that will not harm beneficial species. Apply product correctly. Thorough coverage of the leaves, especially the underside, stems, and branches is essential. The substance must reach the area of the moringa tree where the pest is active. Most failures to control pests result from incorrect application, not product failure. The addition of a spreader sticker to the spray mixture is sometimes suggested when spraying to follow the plant most closely related moringa if you don't see moringa-specific instructions. The use of a spreader sticker will aid in the product adhering to the leaves and improve the coverage for better control. Some formulations of organic biostimulants that are effective in controlling pests are made and prepared with filtration to fit through the tiny holes of the spreader. There should be a sticker on the label that says spreader configuration. Non-chemical control. Sometimes it is possible to remove pests and keep populations below damaging levels by spraying moringa trees with a forceful stream of water. The next best application would be to use a mixture of biostimulant and water solution in a spray. If you don't have biostimulant, use neem oil and water. If you only have water, use a garden hose with an adjustable nozzle and spray the undersides of leaves and stems when such pests as aphids, mealybugs, spider mites, thrips, or lacebugs appear. If the water source is from the tap or city, be sure to have a chlorine and activated carbon filter on the hose. Otherwise, yellowing of the leaves may occur, attracting more pests. Another way to remove pests is to apply diatomaceous earth. It will wash away after a rain or watering, but with a dry application of at least 24 hours, it will prevent the reproduction cycle from occurring, significantly reducing populations of several types of pests. Throw handfuls of diatomaceous earth on leaves and soil as even as possible, about 1 cup for every 5 square feet. Insecticidal soaps and horticultural oils are available that are formulated for controlling insects and related pests. Some people use a small amount of dish soap in their neem oil mixture. An effective, environmentally safe, general-purpose spray against insects or mites attacking moringa trees can be a soap plus oil mixture. Thorough coverage of the underside of leaves is very important because insects must be contacted directly by the soap and oil for control to be successful. Monitor plants and reapply when needed. Commercial formulations of Bacillus thuringiensis, BT, a bacterium, are available and can provide control of some pests such as moths, beetles, and mosquitoes. This bacteria has no effect on other groups of insects, including the predators and parasites. Mechanical control is sometimes practical for small numbers of the large insects such as caterpillars, grasshoppers, and beetles. Frequently, insects can be picked or knocked off the plant and destroyed. Large numbers of newly hatched caterpillars or beetle larvae may be detected by watching for freshly skeletonized leaves and destroyed in this way. After hatching, small caterpillars and beetle larvae remain on the leaf for several days. Damaged leaves can be removed and destroyed before the larvae have dispersed over the moringa trees. Early-stage lubber grasshoppers also feed on moringa leaves and can be hand-picked and destroyed. Chemical control. Chemical pesticides are never required to control insects and other pests on moringa trees. It's not organic if there is a situation where they may be used on other plants, as conventional growing practices with lettuces and spinach. Then there was something missing in the design and implementation of the operation, whether it was planted in the wrong location or timing in the season. There should be some adjustments to the overall scope of the project and find out where the problem may be originating from and test without chemicals. There is no need for chemicals in a thriving ecosystem, which can be achieved with permaculture and regenerative practices. Most chemical control options are designed to kill pests by direct contact, act as stomach poisons, or provide barriers on the plant surface against initial infestations. Some also exert a fumigating or vapor action under certain conditions. They can even move into and through the tissues of the moringa tree if applied. There are products that say they will effectively control the pest without injuring the plant, but that doesn't mean humans and animals could consume its leaves and other material without long-term effects. If you decided to use a non-organic pesticide, using the wrong pesticide in harmful amounts could result in another pest outbreak later, even at another location if the tree is transplanted. It could kill beneficials and other non-target organisms. Before using a chemical pesticide, consider the following points. Select a pesticide that is recommended to control the target pest and may be safe on moringa trees. It may not kill the moringa tree, but it could affect the quality of the leaf and seeds, resulting contamination. Don't use more than the labeled or recommended amount. It could be illegal or may injure the moringa tree. Too little won't control the pest. Certain pesticides have specialized label instructions to avoid phytotoxicity and maintain efficiency. Read the container label carefully. Always check the label to determine whether anything may be mixed with the product. Some pesticide labels specifically warn against mixing certain pesticides together or combining them with fertilizers. Avoid irrigating treated areas for at least 24 hours after application. Systemic insecticides. A systemic insecticide is a chemical compound that is not organic and can be absorbed by the moringa tree. They can be absorbed through the roots and translocated throughout the tree's tissues and makes it toxic to certain insect and mite pests. Could those toxins be harmful to humans if built up in the system over time? Several systemic insecticides can be taken up from the soil by the roots, and they are on the market and used by several commercial growers for conventional moringa farms. Always shop for safer alternatives and grow your moringa organically. In this method, the chemical insecticide could be translocated throughout the tree's tissues. Others can be absorbed by the leaves or stem sprays or from injections into the moringa tree stems. Systemic insecticides have been effective primarily in controlling small sucking pests, including aphids, whiteflies, scales, mealybugs, lacebugs, and spider mites. In general, they have not given satisfactory control of chewing insects. Systemic insecticides applied to the soil as drenches or granules can remain effective over 6 weeks. The residue could leach into the moringa tree, reaching the leaves. Moringa trees can pull heavy metals from the soil. Usually, a test can be applied to moringa trees for heavy metals in the leaves. If they are abnormally high, the use of pesticides is most likely the case. Also, when applied in this manner, they are relatively harmless to any insect predators and parasites that may be presently on or near the tree because they're not feeding on the moringa, but they are absorbed by the tree and passed on through the organic material. Systemic insecticides are available in different formulations and concentrations, and the amounts may vary for different locations. Consult with your local environmental protection agency and follow the directions and cautions on the manufacturer's container label for the amounts to use on other tropical fruit trees as a reference. At the time of application, if a member in the collective uses this method, they can no longer supply orders from that location for the next 3 years. Spray equipment. Spray equipment can be a major hurdle to acceptable pest control in many situations. Most commercial moringa growers, farm managers, and pest control operators have learned there is no substitute for the right spray equipment. Pest control demands uniform application of the control mechanism to the moringa leaves and tree trunks at recommended label rates. Sprayers of various types and sizes are available, from 1 to 3 gallon compressed air sprayers and knapsack sprayers to battery or gas-powered sprayers of larger capacities. Sprayer type varies with the size and type of planting to be protected. With all sprayers, it is important that the leaves and stems be covered to the point of runoff. Sprayers, which attach to the end of the garden hose, represent the cheapest form of application equipment. Although uniform pesticide application with a hose connected to a water source is quite difficult and time-consuming, the spray pattern is usually spotty, and it is difficult to direct the spray to reach and adequately cover the undersides of the leaves, especially those near the ground and on the side of moringa trees close to a building or fence. Considerably more control management will be used with the hose end attachment, and this leads to greater harm to the environment. Solid fertilizer spreaders are excellent for application of granular formulations. A carefully calibrated spreader will properly dispense a granular pesticide across a given area. Even with this equipment, several problems exist in obtaining good pest control. Hydraulic boom sprayers are ideal for large-scale applications. Typical sprayers have a tank capacity of 10 gallons or greater, are engine-driven, and have constant tank agitation to allow use of all organic, non-chemical formulations without plugging. A multi-nozzle boom that is wheel-mounted allows rapid application over large areas. Uniformity of coverage is achieved by a constant boom speed and a constant pressure that is valve-regulated. Calibration of the sprayer for proper rate application is determined by the interaction of pressure, nozzle size, and boom speed. Most of these sprayers are used at a pressure from 40 to 100 psi when applying these liquid pest control mechanisms. Phytotoxicity or plant injury. A pesticide or mixture of pesticides may cause injury to moringa trees. The condition under which the injury occurs may vary considerably depending upon temperature, humidity, and other environmental factors. In general, it is best to apply organic control mechanisms during the cooler part of the day. Moringa trees are less likely to be injured by an application of organic applications, especially when the application timing is protected by at least broken shade for several hours, as opposed to being in direct sun right away. It is a good practice to irrigate moringa trees 1 to 2 days before applying any pest control agent. Some added control mechanisms can injure moringa tree leaves even more when they are stressed for moisture. Using wettable powders, emulsifiable concentrates, emulsifiers, and solvents are all chemical compounds that could cause phytotoxicity and/or injury to your moringa trees. Precautions. Any type of pest control system can cause injury during application. It can be dangerous. Be very careful of your face and eyes. Some organic solutions are still very strong and need to be used with caution. Pesticides are poisons and should not be handled. Read the manufacturer's label of anything you may be using. Before opening the container, observe all instructions and precautions. Wear rubber gloves and other personal protective equipment, PPE, specified on the label when handling and applying any pest control mechanism. Do not spill sprays on skin or clothing. Do not breathe mists or fumes. Wash exposed parts of the body with soap and water immediately after using anything. Store mixtures locked up in original labeled containers and out of reach of children. Rinse empty containers and put rinse water in spray tank. Dispose of empty containers in a manner directed on the label. Pest management strategies. Moringa can be grown just like a lettuce, an intensive annual for biomass and greens, ranging from 10,000 to 100,000 trees on 1 acre. Grow moringa more as a perennial tree at about 400 trees an acre spaced 10 feet apart on a grid. The urban landscape, however, may not have that much space in one place and would need to grow in smaller areas. Believe it or not, growing moringa trees in neighborhoods around the USA is a much more diverse system with smaller areas that includes many different environments for moringa to thrive. It is a complicated system to be managed by humans because, unlike in commercial agriculture, the moringa trees are fragmented and separated by developed areas. This can also be seen as a gift. Pests don't spread as far and wide, which damages crops less. Also, a wide range of beneficials exist to keep the pests from taking hold. Some growers, because they live in the urban environment, keeping up with the Jones, may desire a perfect, well-manicured look, while others may prefer a more natural appearance that requires less work to maintain. Several options exist for managing insects and mites on moringa trees without the use of pesticides. These options include handpicking, knocking, or hosing pests off plants with water and then destroying the pests. Removing infested moringa trees completely, or at least the branches and parts, e.g. pruning, that are infected. Then burn or destroy infected material to reduce the chances of pests moving among your moringa trees. Buying or using moringa trees that are stronger genetically with less chances of mutation are more naturally resistant or tolerant to certain pests, which greatly reduces the need for future pest control. Any mechanical or cultural method that prevents or excludes pests from the location should be attempted before using a pesticide, i.e., permaculture and regenerative agriculture practices. Insect and mite control. A lot of insects, mites, and other insect relatives feed on moringa trees. Many are harmless, some are beneficial, and some are pests. Some pests may need immediate control, especially if present in great numbers, but others may not be worth the time, effort, or cost of control. Pesticides may cause real damage to the moringa trees and could make them look bad or unhealthy. Correct identification of the problem can save money and prevent unnecessary chemical use. After the pest is correctly identified, information can be found on its life cycle, food preference, and habits. It is important to understand these things to properly time any corrective measures. Management of moringa insect pests. Non-chemical suggestions for insect and mite management are listed below. Remember that keeping the plant healthy can oftentimes be the best control method. Recommendations for chemical controls are available in the University of Florida IFAS Insect Management Guide. Ants, scales, mealybugs, aphids, whiteflies, lacebugs, commercial insecticidal oils, and soaps offer effective control of these insects. Thrips control requires insecticides that need to be reapplied 7 to 10 days after a first application. Caterpillars and cutworms. Bacillus thuringiensis will control only caterpillars and will not harm any non-pest species present. A spreader sticker is recommended with Bacillus thuringiensis sprays. Grasshoppers and katydids. Frequently, grasshoppers and katydids can be removed from the plants and killed by hand. Oils do not provide as effective of control, but they usually don't harm the moringa trees too much. If you find there continues to be an issue, there are several predators that eat grasshoppers, like turkeys, that could provide alternative effective control. Beetles. There are very little natural remedies that can ward off many beetles, but luckily they don't harm moringa too badly. An insecticide may be your only alternative if you have continual damage from beetles. Spider mites. Commercial insecticidal oils and soap sprays are effective for controlling mites. Applications must be made several times at 7 to 10 day intervals. Leaf miners. Leaf mining pests are difficult to control because they are inside the leaf walls, protected from oils and treatments on the surface. Systemic insecticides are really the only proven remedy for leaf miners, offering more control, but that could also harm the tree and affect the quality and would not be considered organic from that type of treatment. Borers. Preventing borer attacks by keeping the tree healthy is the best control. Follow all approved cultural and maintenance practices, especially regarding susceptible conditions where they may find a home in moringa trees. Insecticides may be used as directed on container label for control, but then again, the moringa tree products would not be considered organic from that time. Sooty mold. Sooty mold may be washed off with soapy water. Ants. A combination of baits, mound treatments, and broadcast applications of insect growth regulators may be necessary for adequate control. Slugs and snails. Baits are recommended control. Chapter 6.6 Moringa Disease Management. The most important point in controlling a disease is choosing the best control method or series of control methods for a given situation, including permaculture practices that keep the moringa tree roots and soil healthy, reducing stress. The best control method for disease on one type of a plant may not be the best method for another disease on the same or different plant species. Also, the use of more than one control measure, systems control, is often needed. In the moringa tree farming industry, we try to control diseases by finding the part of the life cycle of a living pathogen that could be eradicated at the right time so that available control methods are as effective as possible. We also attempt to use control methods early in relation to pathogen propagation. Once the pathogen increases in numbers, usually accompanied by an increase in disease, control becomes difficult. Most moringa disease control methods are preventive if the moringa trees are planted properly and given ample sun and heat with proper soil conditions. Some eradicative techniques are available for disease control, but it shouldn't ever get that bad or be relied upon for consistent control. Moringa tree disease control. Diseases can often be a result of stress caused by non-living agents, like weather. Moringa trees can't move around when there is something going on like a draft. These non-living disease agents might cause the moringa tree's immune system to weaken, causing the trees to be more susceptible to diseases. Sounds very similar to human conditions. Diseases are often controlled by simply adding vitamins, minerals, and organic fertilizers to the soil. Avoid excess amounts of fertilizer, control the air pollution source, or protect moringa trees from the adversities of the weather in a greenhouse to help prevent disease. Moringa trees affected by non-living agents are often more susceptible to diseases than the actual living agents themselves. Diseases caused by living agents are controlled by different methods. For some diseases, the only way to combat them is through resistant varieties of moringa. Genetically more resilient to diseases are the best means of control. The use of resistant varieties is the best way to reduce the effects of diseases spreading. The difference between profit and an economic setback may depend on obtaining healthy stock. There are several agricultural collectives that follow this disease free seed program, and it follows this same principle of control. Completely removing infected moringa trees or eradication and exclusion have been effective methods for controlling severe diseases. Exclusion of disease is one of the purposes of quarantines. For example, some plants that have diseases have been stopped from entering this country by the quarantine service. Lettuce mosaic virus has been controlled by indexing seed lots. Those seed lots, which contain virus-contaminated seed, cannot be planted in Florida. Citrus canker in Florida was controlled in the early 1900s by both exclusion and eradication. Eradication of disease may be done by other means also. For example, we can attempt to eradicate or reduce a disease by removing other species of plants that are also hosts of the disease. These plants may be weeds or alternate hosts. Alternate hosts support part of the life cycle of the organism causing disease. Removing and destroying diseased moringa trees can also be used in controlling disease. Moringa surgery can be used to control some diseases. For example, a bacterial disease of woody areas called fire blight can be reduced by removing and destroying infected branches. Sanitation around propagating beds, greenhouses, growing areas, and landscape beds is an excellent control measure. Routinely clean and disinfect potting benches, soil bins, arid greenhouse benches. Disinfect tools and equipment, especially if they have been used in areas with diseased moringa material or general debris. After using tractors and implements in an area known to have a soil-borne disease, steam sterilize implements or at least wash off the soil from the tires and implements before using that tractor in other areas. This practice reduces the possibility of carrying the fungus to a non-infested area. Destroy old moringa debris as it can harbor diseases. Wash your hands and forearms thoroughly after handling diseased moringa trees. Replacement and rotation are other methods whereby disease can be reduced. Commercial farmers replace all moringa trees every 5 to 8 years. Moringa trees can be planted in an intensive method as an annual, helping to reduce disease in the plot. Rotate plots every year with other crops, not just moringa, for a much more resilient system. Crop rotation is less effective for controlling obligate parasites that produce wind-blown spores such as rusts, downy mildew, and powdery mildews. The selection of disease-free stock is another good control measure. This includes not only healthy moringa seeds, seedlings, and the cuttings. Control the material and source of your moringa material. Know your farmer and where they get their seeds and how they were collected, harvested, and processed, shipped, imported, and distributed. Know every aspect of the selection to reduce unnecessary diseases from spreading. Control of insects and nematodes often reduces disease when a disease-causing organism is partly or wholly dependent upon these organisms. Insects and nematodes not only act as vectors, carriers of disease, but also their damage can provide an entrance point for disease-causing organisms. Weed control is beneficial for disease control. Weeds can harbor inoculum, interfere with spray deposition, reduce plant vigor, and reduce aeration within crop canopies. Finally, timely applications of chemicals are used to control many diseases at a very last resort, after all other options have been depleted. The most important consideration when spraying fungicides is to have the fungicide on the plant before infection takes place. Most fungicides are protectants, not eradicants. Recently, therapeutic fungicides have been developed. If you have resorted to the use of chemicals, apply non-chemical control methods to maximize efficacy of chemicals. Look into copper as an effective fungicide and still considered organic. Use fungicides with caution, not fear. Read the entire label and follow the directions. Use only approved fungicides on a given area. Store fungicides in a cool, dry place. Store and transport fungicides in such a manner that children or unauthorized persons are denied access. Disease control in moringa trees. Observing spots and patches of yellow or brown in the leaves does not automatically mean the tree has a disease. While moringa injuries or disorders may look like diseases, they are not diseases and should be treated differently. Because diseases are difficult to diagnose, it is often faster to rule out involvement of other factors than to verify the presence of disease. Diseases are the exception and not the rule for most concerns that might become noticeably concerning. Determine if other factors are causing the moringa tree to look sick. You will solve the problem more quickly and avoid applying unnecessary fungicides. An injury to moringa can happen each time it is pruned or harvested, increasing the chances for disease. Continual destructive physical occurrences, such as harvesting the moringa trees too short, too often can cause stunted and awkward growth, possibly forming a disorder. An interaction between the moringa tree and its environment is usually associated with imbalances in regrowth time periods. Examples would be nutritional deficiencies, cold stress, drought or excessive rainfall, excessive harvesting. Again, while these problems may appear to be diseases, there are no pathogens involved. However, these injuries or disorders may weaken the moringa trees so much that a pathogen may attack the plants and cause a disease. If a disease should affect moringa trees, permaculture practices should be implemented first or while fungicides are applied. If a particular home lawn, recreational site, or commercial landscape has a history of developing a particular disease at a particular time of year, then it makes sense to implement permaculture practices to prevent this yearly occurrence. What are the local farmers doing to combat the disease? Permaculture practices are designed to alter the environment to prevent diseases or at least lessen their severity amongst the local populations of moringa trees. They include selection of seed together, much like we are doing as a collective. The collective's seed population is selected from species and cultivars based on your preferences and most local source of seed to the buyer. Disease control is not simple or easy. Since it is usually not practical or desirable to eliminate the moringa tree itself, disease control recommendations are aimed at 1. Altering the environment so it is less favorable for disease development. 2. Suppressing growth of the pathogen. And 3. Decreasing stress on the moringa trees from non-living agents. Implementing an integrated management program that includes permaculture and non-chemical methods is the key to preventing and controlling diseases. There are 3 steps to disease management. First, correctly identify the disease. Second, identify the conditions promoting infection and disease development. Third, identify the management techniques that will alter or eliminate these conducive conditions. Permaculture control practices. Pruning with sharp, clean blades will leave less of an opportunity for infection or fungus to take hold on cuts. The worst thing you can do as a harvester is trim a branch and it doesn't make a clean break, and the bark ends up peeling down, exposing the inner wood. We will go over more moringa management practices in Chapter 7. Covering the wounds with special breathable materials, paint, or even caps can help heal the cuts faster, create a barrier for entry, and create a healthier environment in a space that is continually being used for production. Don't harvest the trees too much or too low all the time. There is a progressive flow that we like to use when trimming. Harvest at optimum height to avoid stress and more susceptibility to diseases. Avoid planting moringa in low-laying areas. It can stay wet for too long in the summer months, which can especially cause root rot. When any disease occurs, cut below it to remove the part in question. It will regrow new leaves and branches, helping it to recover with more tissue available for photosynthesis, resulting in more energy for turf grass growth and the subsequent recovery from the disease. One of the most common questions is, how much do I water them? Irrigate only when the tree is dry and is experiencing light drought stress, curled leaf blades. Then apply enough water to saturate the root zone of the moringa tree. Make sure the application timing is early in the day to allow foliage drying before nightfall. Most fungal pathogens that cause leaf diseases require free water on the leaf or very high humidity to initiate the infection process. Avoid excessively high and excessively low nitrogen fertility, as both contribute to disease development. Use slow-release nitrogen. Remove excess sticks and branches. After you make a harvest, the moringa tree will flush out or bush out, regrowing several new shoots on an existing branch. The key for creating a healthy environment with sun penetrating all the way down to the ground through the moringa tree, remove any accumulation of extra branches. Each branch that is cut should only have 2 to 3 new branches splitting out from the old branch. If there are more than 2 to 3 branches, remove the ones that are crowding the most predominant new green shoots. Correct the crowding to reduce the spreading of pathogens and allowing the inner parts of the moringa tree to dry out. Especially if it's rainy season, they need to have good circulation to lower parts of the trees with little to no excess sticks and branches inside the tree, causing branch crowding. Avoid soil compaction that prevents proper drainage. These areas may have root systems that are deprived of oxygen, resulting in a weak moringa tree. This is also an ideal situation for root rots to develop. Fungicide application. There is a list of approved substances that can be used as a fungicide by the USDA Federal Code of Regulations and should only be used during time periods conducive for disease development. It is acceptable to use those approved substances as fungicides on a preventive basis prior to disease development if you really understand what diseases, pathogens you are protecting the moringa from at any given time of the year. Only use organic and approved fungicides when necessary, as overuse has the potential to increase or shift the disease spectrum or lead to development of fungicide-resistant strains of pathogens. Think about fungicide applicants relative to other maintenance practices like harvesting. Do not harvest for at least 72 hours and preferably longer. The fungicide is probably on the leaf. If you harvest and collect the leaves shortly after application, you have also collected the fungicide. Unless the product is supposed to be irrigated into the soil, do not irrigate for at least 24 hours after a fungicide application. Ideally, the applied area should be completely clear from all other debris and material and well irrigated prior to a fungicide, unless the issue of the fungicide is a result of overwatering. Allow a maximum time interval between fungicide application and the next moringa tree harvest and maintenance operation. Chapter 6-7 Nematode Management. Managing nematode pests on moringa trees. First, it is important to realize that there are a few plant parasitic nematodes in nearly every square foot of soil in Florida. However, since moringa trees are so valuable with their fast growth, available biomass, and nutritional value, they are healthy assets to their surroundings. In most cases, there are several unique environments moringa can grow, depending on the use, but in some cases, they could be planted into any site and end up with serious damages, and it may not be immediate or noticeable until it's too late. With proper site selection and preparation, moringa seed genetics, and continued care during the life of the planting, anyone can establish and maintain an attractive moringa farm operation in Florida, despite the nematodes that infest the soil nearly everywhere. The keys to minimizing the effects of nematodes on moringa trees are good horticultural care and preventive maintenance. Prepare new planting sites properly to give moringa trees the best chance to become rapidly established. Some growers incorporate soil amendments directly into the hole while planting moringa trees in the ground. The effectiveness and health of the roots are highly debated around that subject. However, they generally agree that it is important to dig a much bigger hole than is needed to encompass the root spread or soil ball of the plant, and to provide plenty of loose soil into which new roots can grow. Native soil into which intensive raised beds are to be planted should be prepared well, including removal of any old roots, debris, etc. from the site. Water and nutrient holding capacity of the soil and activity of natural enemies like nematodes will be enhanced by incorporating organic soil amendments into the soil before planting. Feeding the soil also means feeding the nematodes. Treat the soil first. Test to see if high levels of nematodes are present prior to planting your moringa operation. If the plants that are currently on site have been tested and are favorable for nematodes, you may need to remove them. If they were recently removed from the site, treat the soil after they are cleared to give new moringa trees and supplemental plants a better chance of thriving. Some farmers have resorted to chemical treatment or soil solarization, but consult with USDA and Florida Department of Agriculture for approved substances to use and for what case. Testing to see what type of fungus before application is most important in choosing the proper treatment. If there is an infestation after planting moringa trees, the most common chemical used is a triazole fungicide that mostly stays in the stems of the host plants, unaffecting the leaves. Soils may be fumigated by a licensed pesticide applicator only before restoring the location with new trees to reduce nematode populations. However, no pesticide treatment can eliminate all pests from a treated area, nor can a chemical's effects last forever. Nematodes not reached by a fumigant treatment usually reinfest the bed during the growing season, so the soil may have as many nematodes by the end of the season as before fumigation. Soil solarization is a non-chemical way to reduce soil pest populations, but it takes a lot of work and requires the area to be left bare 6 to 8 weeks during the summer. Clear polyethylene is used to cover moist soil that is ready for planting, so the heat generated by sunlight hitting the soil will be trapped and raise the soil temperature high enough to kill many nematodes, fungi, and weed seeds in the upper few inches of the bed. Replace infested, contaminated soil. When fumigation is objectionable or has failed, moringa growers sometimes simply remove all soil plant mix from an annual bed and replace it with new sterile planting soil. Nematodes will eventually invade the new soil medium in those raised beds and bedding. Moringa tree roots can grow out of or under the new soil into the infested native soil, but lining the bed with barrier fabric apparently can delay infection of the moringa trees. Root barriers seem to retard recontamination of the bed from adjacent infested moringa. Fabric barriers will not stop nematodes, but can slow penetration of the moringa roots into the deeper soil that has not been fumigated or replaced. It also prevents growth of infested roots into the bed from outside. Lining an annual bed with root barrier fabric may thus delay nematode invasion of the annual bed. Planting moringa trees in annual beds can be a great way to produce a lot of biomass, but the proximity of the trees can cause more chances for nematodes and fungus to move in. Use nematode-free stock. No matter how perfect and pest-free the planting site is, a nematode infection already started in the roots is right where it will do the most damage. Buy only top-quality moringa trees. Reject any that have clear evidence of nematodes or other hard-to-control pests. Moringa trees are very sensitive to cold weather. The bark is very thin, causing the thin trunks to freeze very easily. But aside from cold weather, moringa trees can adapt to their locations, region, soil type, shade, drainage, etc. Moringa trees that are out of place are more likely than well-adapted ones to suffer environmental stress at any given time, and to be under stress more of the time. Just as you may be more apt to catch a virus if you have not been sleeping or drinking enough water. The cold can place unusual stress on moringa trees. It makes moringa more susceptible to potential pests common in their environment. Moreover, a moringa tree that is well adapted to an area probably has some degree of tolerance or resistance to common pests, such as nematodes. Thankfully, moringa is not as easily susceptible to nematodes. They have a natural protection. The taste of moringa roots is not as appealing to nematodes. Moringa evolved with a built-in pesticide. The flavor of the moringa root detracts many common pests, including nematodes. Do not incorporate other plants that are susceptible to nematodes that are known to occur in or around the site. They will move into the area, making moringa trees more susceptible. Plants vary widely in their susceptibility to different nematodes, just as they vary in form, flower color, and season of bloom. Most references to nematode susceptibility in magazine articles and extension literature refer to one or more root knot nematode species, unless they specify others. Such lists are often incomplete, but it is likely any plant identified as nematode susceptible should not be planted where root knot nematodes are known to be serious. Keep other pests under practical control. Stress from pests can set moringa up for nematode infection just as readily as over or under supply of nutrients or water. Overuse of pesticides can also injure moringa, so use judgment in their application. Give moringa optimum care from the start and for as long as you want them to perform well. Optimum does not mean maximum. Fertilize as needed to maintain healthy growth, not to produce excessive succulent growth that invites attack by nematodes and other pests. Water long enough to deeply penetrate the surface to encourage development of a deep root system that can exploit a large volume of soil for water and nutrients. Frequent shallow watering causes plants to develop a shallow root system. A larger root system can better withstand a small amount of nematode damage without major compromise to the overall health of the plant than can a shallow, already minimal root system. Nematodes live mostly in the top 6 of soil. Do not allow maintenance to lapse. Sudden dry periods or pest outbreaks can weaken moringa in an incredibly short time. Even under normal conditions, erratic or inadequate watering can weaken a moringa so that it can no longer tolerate a modest nematode population that may have existed on with it for years. Mulch the moringa root zone with oak chips to keep roots cool in hot weather and minimize evaporation of water from the soil surface. Organic mulches also contribute organic matter to the soil, thus enhancing the capacity of the soil to retain water and nutrients. Mulches reduce stress on the moringa as a whole and the root system specifically, improving its chances to do well despite some nematode damage to roots. Greater soil organic matter content also stimulates activity of natural enemies, such as certain fungi, predatory nematodes, etc., that apparently help suppress nematode populations. Conspicuously lacking are recommendations of nematicides to treat nematode problems of plants after they are established in the landscape. There is presently no effective nematicide that may be applied legally to moringa already planted in the landscape. Several nematicides that are registered for ornamental plants are restricted to the commercial production nursery because they are extremely toxic. Soil fumigants, the only effective nematicides that can be used legally in most sites, are highly toxic to plants at rates that are effective for nematode control. Therefore, they can only be used to treat soil before planting. Strategies for nematode management on moringa trees. There are several choices for treatment once nematodes are identified as a serious problem in your moringa operation. They are ignore the problem, improve management practices, and hope to live with it. Remove the damaged trees, treat the soil with a chemical that will kill the pests, then replant. Or treat the space with a chemical that will not harm the trees, but is effective against only a few kinds of nematodes. Moringa trees can withstand moderate numbers of most kinds of nematodes with a reasonable level of moringa management. Deep, less frequent watering encourages development of deep roots that can draw water and nutrients from a much greater volume of soil than can short roots, which result from shallow daily watering. Avoid excessive nitrogen fertilization because it tends to encourage growth of succulent roots that will support maximum nematode reproduction. Nematodes growing under such conditions often reach unnaturally high population levels that can destroy roots even faster than a heavily fertilized moringa tree can replace them. Stresses from other factors in the environment should be minimized to reduce susceptibility of moringa roots to nematode feeding. Diseases, especially root rots, are often associated with nematode injury. Nutrient deficiencies and compact soils can make moringa more sensitive to root damage caused by nematodes. When trimmed too often and too short, moringa trees are more susceptible to nematode attack because some of the roots die back at that time, feeding the nematodes. The newly trimmed trees are also unable to manufacture enough carbohydrates for normal root vigor and growth. For about 2 to 3 weeks after a trim, they don't have enough leaves to photosynthesize and heal as fast. It's good practice to leave several branches on a moringa tree untouched. Proper management practices will give moringa trees their best chance to tolerate modest levels of nematodes feeding on their roots. The ability of moringa to grow despite the presence of potentially injurious nematodes can be related to soil composition and thus be affected by soil amendments. Since much of Florida's soil is on relatively sandy soils, soil amendments that improve soil composition may prove helpful for reducing nematode damage to moringa trees throughout the state. Chemical control. At present, the most last resort option to reduce unacceptably high nematode populations in moringa operations is to have an appropriate nematicide applied before the trees are planted. Nematicides may be fumigant or non-fumigant contact chemicals. However, no single product is best for all situations, and none is legal for all situations. The only nematicides available are restricted use pesticides, RUPs, which can only be applied by licensed applicators. Crop rotation, cultivar resistance, biological controls, and other means that are used against pests or nematodes in different crops are rarely practical for moringa tree nematode control. The best approach for most situations is to select a high-quality seed, potentially from the location origin. For example, to get the most genetically resistant strain of Moringa oleifera PKM1, you'll need to get the seeds directly from Periyakulam in Tamil Nadu, India. The effects of nematicides are only temporary. Fumigants have no residual effect, so nematodes survive or escape the treatment. Nematodes that were too deep to be reached by it, or were protected inside intact root tissues, or were even brought in with soil used to complete the grade after fumigation, or in new soil, can begin to recolonize the root zone immediately. A non-fumigant nematicide applied to living tissue may remain in the root zone for several weeks, but will eventually dissipate because of the combined effects of leaching and natural degradation. Those products do not necessarily kill all nematodes exposed to them, but inactivate or paralyze many of them. Therefore, when the chemical is gone, there are usually some nematodes ready to resume feeding and reproducing. Thus, treatment with either type of nematicide provides only a limited period of relief from nematode stress. Chapter 6.8 Weed Control. There are several methods of weed control, generally mechanical control, crop competition and rotation, mulching, weed barrier fabrics, biological control, fire, and chemical control are used to control agricultural weeds. This section focuses on those that apply to the nursery and landscape industry. Best weed control is usually achieved by a combination of 2 or more methods. If effective weed control has been achieved, methods of prevention should be used to keep weeds from reinfesting an area. Cultural methods. Weeds in moringa could be managed to a large extent by cultural practices such as crop rotation, intercropping, mulching, etc. Crop rotation. Certain weeds have association with moringa crop, and hence, monocropping of moringa may lead to severe crop weed competition. Inclusions of green manure crops or fodder crops like sorghum not only suppress weeds, but also help in crop diversification. Tillage. Tillage and cultivation are the most traditional means of weed management in agriculture. Both expose bare ground, which is an invitation for weeds to grow. Bare ground also encourages soil erosion, speeds organic matter decomposition, disturbs soil biology, increases water runoff, decreases water infiltration, damages soil structure, and costs money to maintain for fuel and machinery or for hand labor. Some specific tillage guidelines and techniques for weed management include the following. Pre-plant tillage, where weeds such as quackgrass or Johnson grass exist, springtooth harrows and similar tools can be effective in catching and pulling the rhizomes to the soil surface, where they desiccate and die. Dissing, by contrast, tends to cut and distribute rhizomes and may make the stand even more dense. Inter-row cultivation. It is best done in between the rows of moringa as soon as possible after precipitation once the soil is dry enough to work. This avoids compaction, breaks surface crusting, and catches weeds as they are germinating. The most vulnerable stage, flame weeding. Pre-plant, pre-emergent, and post-emergent flame weeding has been successful in a few crops. The pre-plant application has commonly been referred to as the stale seedbed technique. After seedbed tillage is completed, weed seeds, mostly in the upper 2 inches 43 of the soil, are allowed to sprout. Assuming adequate moisture and a minimum soil temperature of 50 OF to a depth of 2 inches. This should occur within 2 weeks. A fine to slightly compacted seedbed will germinate a much larger number of weeds. The weeds are then seared with a flamer or burned down with a broad-spectrum herbicide, preferably when the population is between the 1st and 5th true leaf stages, a time when they are most susceptible. The moringa crop should then be seeded as soon as possible and with minimal soil disturbance to avoid bringing new seed to the surface. For the same reason, subsequent cultivations should be shallow, less than 2 inches deep. Intercropping. Moringa is planted in wider row spacing. This provides better opportunity for weeds to emerge in a large numbers and infest crop. Inclusion of short duration and quick growing intercrops in these row spaces can suppress weed growth to a great extent. From the 2nd year onwards, moringa can be intercropped with maize, sunflower, and other field crops. Sunflower is particularly recommended for helping to control weed growth. However, moringa trees are reported to be highly competitive with eggplant, Solanum melongena, and sweet corn, Zea maize, and can reduce their yields by up to 50%. The net present value, NPV, benefit cost ratio, BC, and relative net return, RNR, were higher with Cymbopogon and Stachytarfeta herbs. Bryan, 2004. Cover cropping. The cover crops that have shown allelopathic effects on weeds include sunflowers, sorghum, and rapeseed. Weed control ability varies among varieties and management practices. Sweet potatoes have been shown to inhibit the growth of yellow nutsedge, velvet leaf, and pigweed. Field trials showed a 90% reduction of yellow nutsedge over 2 years following sweet potatoes. This can be better utilized by growing in the interspace of moringa. In general, typical levels of cover crop residues, when left on the soil surface, can be expected to reduce weed emergence by 75 to 90%. As these residues decompose, the weed suppression effect will decline also. Residues that are more layered and more compressed will 44 be more suppressive. Small-seeded weeds that have light requirements for sprouting are most sensitive to cover crop residue. Larger-seeded annual and perennial weeds. Mulching. The principle of mulching is to exclude light from the tops of the weeds until the reserve food supply in the roots is depleted and the weeds starve. Mulches include clean straw, hay or manure, tar paper, sawdust, and black plastic. When the vegetation under the mulch has been destroyed, the resultant bare patch must be reseeded with competitive vegetation to prevent new weed introductions. Black polythene mulching. Mulching is an age-old practice, especially in horticultural crops in many countries. Though various natural and synthetic materials are suggested, black polythene mulching is found predominant as it is reported to be very effective in control of weeds in vegetable gardens. Photosynthesis of weeds is arrested and they ultimately die as the sheet totally blocks the sunlight. Further, black polythene mulch increases soil temperature as well as conserves the soil moisture by preventing the evaporation of water from soil surface. The method is eco-friendly and effective against all types of weeds in moringa. Trash mulching, it can be practiced soon after emergence of moringa to 12 cm thickness. Trash cover restricts sunlight and checks weed emergence. Besides suppressing weeds, trash mulching also conserves soil moisture and provides a potential source of organic matter. Mechanical control. One type of mechanical control is burial. This method is most effective on annual weeds in which all the growing points can be buried. Burial is usually less effective on perennial weeds that have underground stems and roots and are capable of regrowth from these underground storage organs. Another method of mechanical control is cultivation. The main objective in cultivation is to cut the root system of weeds. Deep cultivation may bring more weed seed to the surface where they will germinate. Most studies have shown that when weeds can be controlled without cultivation, then there is no advantage to cultivating. In fact, there may be disadvantages such as drying of the soil surface, bringing weed seed to the surface, and disturbing the root system of desired moringa trees. Mowing is another method of mechanical control. Mowing is usually most effective on tall-growing annuals and not as effective on short-growing plants or perennials. The growth habit of the plant usually indicates how effective mowing will be. Because grasses grow from basal meristems, mowing is not usually an effective method of controlling these weeds. Perennial weeds, which regrow from underground storage organs, require frequent and usually long-term mowing for control. This is because the leaf area must be continually removed in order that the underground plant parts are starved and are not allowed to regenerate any carbohydrates for food storage. Annual weeds are usually mowed to prevent seed production and to allow the crop a better competitive advantage. Crop competition. Crop competition is usually one of the cheapest and best methods of weed control. However, it is often one of the most overlooked methods. Competition is especially important in the establishment of thriving permaculture and polycrop systems. Weeds compete with desired plants for space, light, moisture, nutrients, and carbon dioxide. Usually, if the area is cleared from weeds and the moringa tree is planted in ideal conditions, will have the competitive advantage. Therefore, it is extremely important that moringa trees be planted with the competitive advantage. Factors such as planting date, row spacing, seeding rate, planting depth, soil moisture, soil fertility, and soil pH have an influence on the competitive advantage moringa will have over the weed. The best way to choke out weeds organically and in the lines of most natural systems would be with nitrogen-fixing ground covers like mimosa, perennial peanut, clovers, alfalfa, and low spreaders like cowpea. Biological control. Biological weed control as a practical tool has not been used to a great extent in controlling weeds. There have been certain instances of successful biological control programs. However, these have been infrequent. This is an area in which present and future research appears promising. Insects, disease, and nematodes do naturally suppress the growth of certain plants, and this is a continual process in the field. One area which should be recognized is how the use of fungicides, nematicides, and insecticides influences weed populations. Mulching. For weed control, it may be more accurate to say that a mulch suppresses weeds if the mulch is applied deeply enough, at least 3. The application of mulch prevents many weed seeds from germinating, and it smothers small weeds. A large variety of materials may be used as mulch. The type of mulch used is not as critical as the amount. In Florida, yard trash, leaves, grass clippings, and prunings is plentiful, and it is being used increasingly as mulch. These materials are also being composted commercially and are becoming more available in the state of Florida. This is an excellent way to both reduce weed problems and keep these useful materials out of our shrinking landfill spaces. Woven weed barrier fabrics. Woven fabrics are available to use as weed barriers. Because they are woven, they allow air and water to penetrate to and from the soil surface. They, like mulches, are effective in suppressing weeds and reducing maintenance, although some weeds will continue to penetrate the fabric. Soil and weed seeds also blow over the top, where the weeds can eventually germinate and grow. Where fabrics are used, they are usually used in combination with mulches. Neither should be thought of as permanent weed control. They must be maintained and supplemented eventually with repeated applications of mulch or layers of barrier over one another. Chemical control, time of application. Chemical control can be administered prior to planting moringa trees, pre-emergence and post-emergence. Pre-planting refers to applications made before the moringa tree is planted. In most cases, these materials are incorporated into the soil and are called pre-plant incorporated treatments. The great advantage of these incorporated treatments is that the herbicide is placed in the zone where weed seed germinate and is not dependent on rainfall to move the herbicide into this zone. This treatment adds to the cost of incorporation and requires that the crop be tolerant of the herbicide since they will be in contact. Pre-emergence treatments usually refer to applications made after the moringa tree is planted by seed and or established with a small seedling, but before the undesired species emerges. However, strictly speaking, pre-emergence may apply to other situations. It may be pre-emergence to the crop, pre-emergence to the weeds, or pre-emergence to both moringa trees and weed. These pre-emergence applications are usually applied to the soil surface and require rainfall or irrigation to move the herbicide into the soil. If the herbicide is not moved into the soil where the weed seeds are located, it will not be effective. If left on the soil surface, these herbicides are often lost due to photodecomposition and vaporization. There are USDA certified organic herbicides that are safer to use than others. Post-emergence treatments are applied following emergence. However, as with pre-emergence, it should be specified as to post-emergence of the moringa trees or weeds. If the crop has emerged but no weeds are present, then the application is post-emergence to the crop but pre-emergence to the weeds and would be applied to the soil surface. If the moringa trees have emerged and the weeds have emerged, then the application is post-emergence to both weed and crop and would be applied to the foliage of the weeds, not the moringa trees. Chemical control area of application. Chemical application for nursery and landscape plants may involve broadcast, directed, or spot treatment. As the name implies, broadcast applications cover the entire area. These treatments, while requiring the largest amount of chemicals and highest cost per unit of area, usually result in best weed control. Directed applications are applied to a particular area or part of the moringa tree. These applications are usually directed to the base of the trunk away from the leaves. The ability to use directed sprays usually depends on the height differential between the desired plant and the weed. Directed sprays are very useful in late-season control of weeds and usually follow a pre-plant or pre-emergence application. In many cases, pre-plant or pre-emergence applications do not persist long enough to control late germinating weed seed or may not be used on certain soil types. Spot treatments are used for weeds, which are localized in certain areas but are not widespread over the entire area. When only isolated areas of weeds are present, this is the cheapest and best method to control and prevent their spread to other areas. Prevention. If effective weed control has been achieved using the previously discussed methods, one further step should be considered: preventing weeds from reinfesting the area. Knowledge of how weeds enter the field is important. Weed seed may be distributed in crop seed, hay, straw, by wind, water, animals, machinery, and other ways. Herbicides. General herbicides may be classified in several ways depending on where and how they are applied and their action in or on the plant. Herbicides may be either foliage applied or soil applied. They may kill by contact or may be translocated through the moringa trees. Herbicides may also be selective or non-selective. Some herbicides may be effective either foliage applied or soil applied. Whether an herbicide is selective or non-selective may depend on several factors, such as being able to identify the desired moringa trees and only affect the present weed, time of application, and rate of application. Foliage-applied herbicides. Foliage-applied herbicides help to control unwanted weeds in small, intensive beds of moringa trees. They may be applied to the entire area, the leaves, stems, or shoots of the moringa, to kill any vegetation that is less desirable, like weeds. They could choke out young moringa trees. Although, if you plant the moringa seeds close together, they will sprout and for the most part keep unwanted grass and weeds to get root and become taller than moringa. The longer moringa will have to grow without weeds coming up in the same space. The more likely the moringa trees will shade out the weeds and prevent them from taking over. Herbicides that kill only those parts of the plants that the spray touches are called contact herbicides. If the herbicide touches a leaf, it may be absorbed into the moringa tree and moved throughout the other parts, potentially harming it or killing the moringa tree. These are translocated herbicides. For example, if a drop of paraquat were applied to the leaf tip of a young tomato plant, then only that leaf tip would be killed. However, if a drop of 2,4-D were applied to the leaf tip of a young tomato plant, then other areas of the plant would express symptoms due to translocation of the 2.4-D throughout the plant. For these herbicides to be effective, they must enter the weed, and this entrance may be influenced by factors such as the shape or orientation of the leaf, roughness of the leaf surface, pubescence on the leaves, presence of wax, or the formulation of the herbicide. For example, it is difficult to obtain good coverage of weeds with narrow, upright leaves, such as wild onion, since the herbicide bounces or runs off, while plants with prostrate leaves, such as Biden's alba, are much easier to cover. The presence of pubescence, hairs, on the leaf may result in an herbicide not reaching the leaf surface, but remaining suspended on the hairs until evaporation. In such cases, the addition of a surfactant may be useful in reducing the surface tension of the water droplet and allowing it to spread through the hairs onto the leaf surface where it may be absorbed. On waxy leaf surfaces, the formulation of the herbicide may be important. For example, the amine formulations of phenoxy herbicides are not highly soluble in wax and are not as effective on moringa because they are hydrophobic and repel water droplets with their waxy leaves. So using an ester formulation, which are soluble in wax, would affect the moringa way more than an amine formula. Contact herbicides are more effective on annual weeds, but are not usually effective on perennials. Translocated herbicides may be effective on annuals or perennials. Soil-applied herbicides. Soil-applied herbicides primarily enter moringa trees through the root system, and many factors influence the effectiveness of soil-applied herbicides. Factors such as tolerance of the moringa tree. In this case, we want to target weeds for an effective herbicide. Notice the depth of the weed, amount of seeds on them or on the ground, the amount of sand, silt, clay, and organic matter of the soil, amount and time of rainfall, and temperature are a few factors which may influence effectiveness. Persistence of herbicides is extremely important in the duration of weed control and in determining what species can be planted later in the season. The persistence of herbicides applied to the soil may be influenced by numerous factors such as microbial decomposition, chemical decomposition, absorption, leaching, volatilization, and photodecomposition. Soil type is extremely important. Soluble herbicides applied to sandy soils may rapidly leach out of the zone containing weed seed and into the intensive raised bed zone of germination or roots of larger moringa trees, thus resulting in poor weed control and damage to the crop. In fine textured soils or organic soils, herbicides may be so tightly absorbed that they are not available for weed control, or they may be released so slowly that they are not in a concentration great enough to kill the weeds. This lack of release may also increase the presence of herbicides inside moringa trees for extended periods. You don't want to apply an herbicide and harvest right away, and you don't want a lingering herbicide in the leaves when it's time to harvest. Herbicides which specify incorporation into the soil may do so to improve the contact of the herbicides with the weed seed and to minimize the loss of the herbicides by volatilization and photodecomposition. Some herbicides, if not incorporated soon after application, may be lost from the soil surface. One of the major factors influencing herbicide persistence in the soil is microorganisms. These organisms may use the organic herbicides as a source of carbon for their food supply. The specific herbicide microorganism present, temperature, water, oxygen, and mineral nutrients are all important in the persistence. Generally, Fast degradation of herbicides is achieved in warm, moist, well-aerated, fertile soils at pH ranges favorable to 6.3, 6.5 for moringa growth. However, the greatest persistence of herbicides may be expected in soil that is cold, dry, poorly aerated, and generally unfavorable to the growth of microorganisms that contribute to chemical breakdown.